Today we're going to talk about
growing with fishes. Growing with fishes.
Hey everybody, welcome to the Growing with Fishes podcast, episode 287. We're back again with one of our favorite guests on the show, Kevin McKernan. Thanks a lot for joining us tonight.
Hey, thanks for having me. 287 is a big number. Good for you guys.
Oh yeah. No, I still look back and think about how Marty and I having a disagreement on Facebook turned into 800 hours of educational content on cannabis, but it certainly did.
And you haven't been banned. That's amazing.
Right? I mean, we've come close. There's twice that we've come within one strike of losing the channel. So it's not for lack of trying. I'll put it that way.
Yeah.
Oh, man. We also have Fumidor with us today. Thanks a lot for joining us, Fumi.
Howdy. Cheers, guys. Happy to be here. Nice to see you.
Where can people find you real quick before we get started?
I have a website, Fumidoro.com. Honestly, is the best way. You can pick up some interesting genetics over there, but also we have a couple of fun podcasts per week. Basically, we just had one yesterday over at my channel, Fumidor and the Flavors, over on YouTube. So basically, change the channel ever so slightly from Potent Ponics when it's over to Fumidor and the Flavors, and you'll see some good content from us talking to Coot and talking to Potent and Jinx Proof a couple weeks ago. Lots of good stuff. So thanks for asking. Cheers.
I think you might actually have more hours of content than even I do.
It's possible because we have those long shows. Yeah, 100%.
Yeah,
5-6 hour shows sometimes, straight up.
Well, you guys can check Marty and myself, who's the normal co-host of the show, who wasn't able to join us this evening, over at APMJClass.com. We do have a long format Aquaponic Cannabis class. If you're interested in learning more about aquaponic cannabis from top to bottom, hash making, pest control, nutrients, planting, all different types of hybrid systems, and everything else you can think of, be sure to check that out. We have over 700 slides and lectures individually on that class. So be sure to check that out if you're looking for that type of info. You can use coupon code AP420 in order to save $75 off the class now through about 28 hours from the recording of the show at the end of Earth Day. So if you're looking for a discount on that, now's the time. We only do two big discounts, one for 420 and one for Black Friday. So definitely check that out if you're looking for a little bit cheaper access to aquaponic cannabis knowledge. We also have a new aquaponic cannabis class. I'm sorry. My brain is a bit fried today. Too many hours in the sun. You can check out our new aquaponic. I can't even talk tonight. Our new cannabis class out in Oklahoma City with Jordan River and myself from Growcast. We're going to be doing a regular monthly grow club out there. So Jordan and I are going to give little local grow classes on, how to grow at a home scale. We're not going to make it anything about commercial, but we're going to have a little bit of a monthly meetup. So if you're looking for seeds and you're in the Oklahoma City area, or you're looking for more just general information for, a new community to hang out with, definitely check us out. All right. Well, thanks a lot for joining us, Kevin. I know we were hoping to hear from you earlier this year, or I guess it was the end of last year, but you were quite sick there for a little while, but we're happy to have you back. And thanks a lot for joining us tonight. I know you have a ton of amazing stuff to tell us about tonight, and you launched a whole new project yesterday, which we're going to get to in a bit. And you have a whole amazing conference that you got going on next week and all kinds of other amazing stuff. You're truly one of the leaders when it comes to open source knowledge. And I think kind of leading the way and the way that the genomics sector needs to be in terms of open source and all the rest. So thank you so much for joining us tonight.
Oh, thank you. Yeah, I'd love to share some of the info with you. Yeah, we did just release I think there's 84 genomes now. So this is the psilocybe genomes that we put public on 4/19. And then we did some stuff that's actually probably worth chatting about in your community as well with the Humboldt Grace Project. Legacy project. I think they're doing some work on building like an auction house that's run on blockchains and a way to secure smart contracts and IP rights on people's genetics. And if there's sequencing involved, there's that angle as well. So yeah, 2 big days. A lot of things announced on on 419 and 420. It's hard now that you're in both spaces. You have to prepare for both days, and they're back to back.
Especially if you're into organic chemistry, it's quite a busy week.
Yeah.
As someone who's equally as passionate about organic chemistry as you are, there's quite a few different things. A lot of people don't know that not only did Albert Hofmann discover LSD and lysergic acid, but he also was the first person to synthesize and isolate psilocybin and psilocybin, and the person who administered chemical-derived psilocybin to Maria Sabina and took it with her to see if she thought there was any difference. So he really did play out. And not only that, but I mean, the Hoffman degradation. And I mean, the guys literally played such an incredible role in the history of many things that a lot of us are passionate about.
Yeah, no, indeed. That's it's fascinating history as well. And there's been some progress on people actually cloning some of that pathway into yeast. I think Hofmeister's lab did that. And so they can get yeast to brew this stuff now. I think that's been giving it a particular L-tryptophan precursor. And then I don't think as much has been done yet on the beta-carboline pathway, which is something that is, a paper came out demonstrating that some of the mushrooms also make those, and they're MAO inhibitors, so they slow down the metabolism of psilocybin and presumably increase blood levels of those drugs. So, and I think one of them is actually, they believe is psychoactive as well. So there's that whole side of the pathway. We just don't know what genes make those. I think we have good understanding of the genes that make everything on the psilocybin side of the pathway, but what we're trying to chart out with some of these genetics is, are there any variants in these genomes that might predict things that might arrest along that pathway? Very, very analogous to CBG moving its way into CBD or THC. There's a precursor here, and on its way to becoming psilocybin, there's a couple stops on the way that could make the other compounds, originacin, baeocystin, and norbaeocystin. So there's an entourage effect we got to keep our eye on in this thing.
I've never really thought about that, but you're absolutely right. I mean, especially with things like copy and tetrahydroharmaline and some of the other things, there's been no studies done on any of the genetics around amplifying compounds or any, some of the other things that are synergistic with a lot of the things that people traditionally understand as medicine or medicinal compounds.
Yeah, the thing I did learn digging into some of this literature is that psilocybin is mostly made in the fruits, the carbophores, and the beta-carbolines, they seem to be 100-fold higher in the mycelium. So there may be a class of genes that are related to both of these that are differentially expressed in different tissue types. And maybe that, I don't know, we need to do some RNA sequencing to sort of figure this out and see if there's certain differential genes that are expressed that kind of highlights what areas we should be looking at a little bit more closely. Those, of course, a little bit more difficult to do these days because getting RNA out of live fruit is naughty, at least in where I live. So it has to probably be done in Canada or somewhere else.
It's really interesting because so many of these compounds have had such an incredible effect on people's lives, yet we know so little about them on a genetic level. And I think that it's almost comical that how little we understand about some of those things. And then also just how critical the work that you're doing in terms of putting these sequences into the open source kind of setting, and preventing these from being locked down by various nefarious entities that we've seen attempted so many times in the cannabis side.
Yeah, I think some of that's already happening. I heard there are some patents on the application of psilocybin for depression that might go back to Compass. So yeah, that game's playing itself again. Did you guys see that? I'm curious of your take on the DEA announcement recently about type 1 cannabis seeds. There's some news about that, which is a bit of a departure.
Yeah.
A very huge departure.
So that we've always been going under the more stringent guidelines of the Hemp Industry Association versus the DEA, where the DEA was pretty adamant about it can't be viable tissue. So we've always been having people isopropanol treat anything that they send. First off, no leaves, no flowers. Send us, if you do sequencing, send stems that have been isopropanol treated that strips the cannabinoids off. It also fits the bill of making it non-viable. But now they've kind of stripped that viability issue And I'm wondering if that sets a precedence for psilocybin, because obviously the spores have been legal for decades and no one seems to have gotten any trouble selling these spores. I know one or two cases maybe where they were selling and growing and that crossed the line, but the spores seem to have gone across state lines quite readily.
I think the real trial case would be to see if peyote seeds, because peyote and cannabis are the only two species that are banned by the DEA. Unless you can correct me on, and you know of another one.
Yeah.
Those are the only two specific species that are banned by the DEA. So, you know, does that nullify that across the board? You know, can you purchase peyote seeds and for peyote buttons, you know, in a seed form, as long as it's not clonal tissue? You know, and the other question is that the way that they ruled that seed ruling, like, does that also apply to clones?
Because I know that's what I was.
I think some 0.3% or higher. So like,
yeah. I think some people are interpreting that way, and that does certainly change the dynamics of like the clone market, and I think it also is going to change the migration of pathogens. Obviously there's a lot of HLV or hop latent virus out west.
There's scarier shit than that, I'll tell you personally, and I'm happy to throw some pictures up of it. And I've shown this on Fumi Show a couple of times. There's as far as my end, again, this is all anecdotal. And we preface everything I'm about to say with, this is strictly based on my personal observations at 3 different grows.
No, we're going to cancel you if it's not peer reviewed.
Oh yeah, I'm so sick of that shit. But I have observed a cucumber beetle, as far as I can tell, cucumber beetle vectored virus or bacteria that melts plants in 2 to 3 weeks. But we've not been able to get any type of ID on it. It's something that I'm very much interested in sending off to you, but this is what it does to the bottom of the leaves. And I sent some of the pictures of this stuff to every pathologist I can think of.
Oh, wow.
In terms of mosaic patterning. But every plant that got this particular disease was attacked by cucumber beetles. So I was very curious on.
There is a cucumber mosaic virus. I think we've had our eyes on. I wonder if did you know if it's virus or bacterial, or is it?
I'm not sure. All I know is that here, I can show you the top of that same leaf here. Hold on. Where is it here? So here's the bottom of the leaf and here's the top of the leaf. So here's the bottom and then here's the top. The top does not look anywhere near as bad as the bottom.
Yeah.
That's the weird thing about it, is that it has this weird one-dimensional patterning.
Have you scanned it for HLV?
I have not. These were all, the other reason is these plants had both spider mites and cucumber beetles and outdoor sitting in Oklahoma. So like, not a really limited choice of vectors on that one. And it was outdoor, right? So
I mean, there was a study that came out of Colorado on beet curly top virus. I mean, the odd thing is a lot of these mosaic things, when we first saw that on some plants out here that Colin Palmer showed.
I have good pictures of beet curly top that have been confirmed. Hold on. Give me 2 more seconds. I can pull those. 1 second. So these were from a confirmed beet curly top virus infection. Hold on. Wrong year. I'm gonna go to 2019. Hold on. I got some really good pictures of those ones too. A buddy of mine ran into a bunch of that in Oklahoma. I'm sorry, Oregon. Here's beet curly top. For those of you that aren't aware at home.
Oh God, yeah, that's unique.
You know, there's really strange curling.
Yeah. Yeah, not a lot of mosaicism there. It's just the curling of the leaves.
Yep.
We have seen that mosaic pattern though in some hop latent viral infections, which is it wasn't in all of the plants, so that's what was kind of bizarre. Some of them would give you that. It was a little bit more extreme chlorosis than what you're seeing. Yours just looks like it's going a little bit like, you know, an albino or what have you. It's not as yellow, but I
have some more progressive pictures on that on my. I think they're on my phone and not on this account on the computer, but yeah, there's quite a few different. I've seen that in the mosaics. I've seen probably, in my opinion, 4 different mosaic species that have consistent patterning that's unique to that, variation of whatever, either be it strain or species. At least in all the years that I've done it between, all the things. And I think that just like you're saying, the cucumber, alfalfa mosaic, and we're going to find more of them that are jumping over.
Yeah. And the interesting thing is they're not always the same sequence as what you find in the plant that it might be named after. So there's tobacco mosaic has been thought to be in cannabis. I've heard recently from Punja that he's found it on cannabis, but it's diverged enough from the initial one to probably require unique primers for it, at least in the PCR front. But it is getting picked up by an ELISA. So there's that one's finally checked off. The longest time we were like, we've just not seen it in the literature yet. It's but it's different. And this what really I guess this isn't too surprising given these things are RNA viruses. But there was another study out of Colorado, and that person was on the CannMed podcast, Coffee Talk, talking about what they had found. And their tobacco streak virus was only like 83% identical to the one that's been found in tobacco, and also needed some adjustments in order for them to properly pick it up. So I think what we got to do is take a plant like that. And, the first thing to screen it for is probably do an RNA-seq analysis to see if you can find novel viruses. And if you don't find RNA viruses, you then have to go looking for DNA ones, which are a little perhaps easier. But and then, of course, try to get the microbiome out of it, because it could be a bacteria or fungi. But it's not always easy to get those off the cannabis plant because it comes with all of this background cannabis DNA. So you're mostly sequencing the genome of the plant and hoping to find some reads that represent the microbiome. And you usually find more than one microbe there. So you don't know which one is the causative agent unless you can maybe culture it or re-inoculate onto some other plant and go through that whole Koch's postulate approach. But it's not a simple one and done single test tells you the answer on those things. It's really an R&D project to go and discover what the hell it is. And is it really widespread such that it's worth like developing tests for it that would track it in more than one place?
Sure. You just mentioned the CannMed podcast. Tell us a little bit more about that, and more people can find it.
Oh yeah, it's called Coffee Talk. I think it's on iTunes. Ben's been running that out of MGC, and it has a great list of guests up there. They're usually guests that are about to be speaking at CannMed or have spoken at CannMed. So there's a tie in there, but it spans the gamut. We get a lot of analytical chemists on there. We get folks speaking about all these viruses, tissue culture people, the whole and all the way to folks, physicians treating people with varied cannabinoid profiles. So worth a listen.
Yeah, I didn't even realize you had a podcast as well, or I would have been listening in this whole time, because you're definitely a
yeah. Oh, I don't run it. I'm lucky if I get on it. I don't know the time of the day. I wish I did. But all that credit goes to Ben. You'll meet him at CannMed.
So tell us a bit more about CannMed and some of the awesome talks that you have coming up for this year.
Yeah, so it's going to be a great year because it's been like 2 years of buildup. We kept trying to have this thing, and it kept getting COVID canceled because it was in California, and of course California was a little bit more strict than other places. But Pasadena was still not allowing us to have it, or they were allowing it to be done with all types of discomforting things of like masking and vaxxing and testing and all this madness. So we just kept punting it because we felt like that would be a miserable experience. But finally, it's happening. So there's a host of folks. I think there's going to be some discussion there about this legacy, this Humble Grace Legacy Project that Lele has been organizing. They'll hopefully lay out the whole infrastructure of that auction system. I'll be giving a presentation on some of the psilocybin work that we're doing. Yvonne's giving some presentations on some of the pathogen detection things we've been doing the last year. There's a website that has a list of the schedule should be up there now. So there's spreads the gamut. I think Seth Crawford's probably going to be there talking about triploids and whatever else they're up to lately. I'm having a bad time recalling everything from memory because I remember reading these abstracts 2 years ago, and I'm sure whatever people are speaking about the next week is probably diverged from the abstract they sent us by 2 years. So they're somewhat placeholders, but it's long overdue. We've been missing this kind of in-personal conference. That's these Zoom things are as fun as they are. They're not the same thing as hanging out with everybody and sharing notes.
For sure. I know Fumi and I went to the first two Regen conferences this year, and it certainly was a huge difference hanging out with people in person versus digital format in any time.
Yeah. It's hard to. A lot of these conferences just stopped during the pandemic, so it's very much slowed down the rate of progress in many ways. These things rarely get considered when we're putting the brakes on society, but the rate of progress is definitely about human interaction, and that's certainly been attenuated in the last few years. So we're looking forward to getting it going again.
Oh yeah. So I wanted to jump quickly back to your Cilacida project. You want to tell us a little bit about that, and then like some of the different things that you've discovered? Because I've certainly never seen anyone take even half as much of the time to, or not even a fifth of the time to study the genetics of that particular thing. And I think that you're really kind of leading the way in terms of the genetic understanding of what's going on with those types of fungi, and then also the expression of all these different secondary compounds, which I think that the more we learn, it's going to end up being like cannabis, where all these secondary compounds are having a much larger effect on the medicinal aspect of it than we initially realized.
Yeah, so this was a bit of a lockdown project. Many years ago, I actually sequenced one of these genomes when my father was sick with cancer, but I was a little afraid to put my name on it because I was making cannabis oil for him, and I didn't want anyone knocking on my door because of putting this sequence public. But so I tried to put that. I think I handed some of that to MAPS, and I put some of it public up on semi-public up on some blockchain system called Alexandria that kind of unfortunately disappeared. And an open bazaar, I think we had a link to it. So it's not really been Google crawled very well. So I don't think people find it very readily, but we finally packed that up and put it into NCBI, and then decided it's time to we should sequence many more of these, mainly because there's all this literature coming out that fluvoxamine was working for COVID. And fluvoxamine's an SSRI, but it's unique in its SRI properties. It doesn't just hit the 5-HT like serotonin pathway. It seems to be playing a role in the sigma-1 receptor pathway as well, which is somewhat unique in its properties for SSRIs. And that's a receptor you may know from DMT. There's papers showing DMT hits sigma-1 and obviously 5-HT2A, but I don't know, that got us intrigued that, hey, there's a mushroom out here that makes these things. And if I had to bet, psilocybin would probably behave a lot like fluvoxamine with probably lower side effects. Certainly third rail, but why not sequence these things to better understand these compounds? If psilocybin works, maybe there's another compound in the plant that's a couple methyl groups different that might be less psychoactive and still get the same desired antiviral effects that they're seeing. And so there has been work done on this before us. And there was, I think, a group called Hofmeister. There's another, Jason Slott's done some work in this area. I think Byron Dinginger's done some work in Utah. There's other folks who have been sequencing these, usually cyanescens or cubensis, one or two genomes at a time with Illumina. But to really do these things right, we wanted to get a reference sequence done that was rock solid. And so we made some high molecular weight DNA and called up Seth. We were going to send it to a university, but during COVID, like all of them were backlogged and couldn't really give us any guaranteed turnaround time. So we asked Seth if he had any extra space on his sequencer up there, and he did. So he threw this high molecular weight DNA through there and got us a really great sequence assembly of the penis envy genome. And then once we had the raw data, like a bunch of other assembly people were like, "Oh, let's all try this." So Jason Chin assembled it with Peregrine. I think we ended up using that assembly, but we also had High Canoe assemble it, Sergi Koren's group. We used his code. I don't think he did it, but so we had a couple assemblies out of the gate. That resulted in a paper we put through F1000 that had this gorgeous reference genome. And Phase Genomics was very interested in helping out as well. So we got them some spores, and they were able to basically get us a high C map of this thing, which is, so I think I've described this in one or two of these other talks, but when we got the first sequence back from the PacBio Hi-Fi system, it ended up in about 40 or so contigs, which is really good for a genome. Like 2 of the chromosomes were in perfect tip-to-tip sequences, like 3 or 4 million bases long. We had telomeres on both ends. It was like, you just don't. This is not the way genomics used to work. Like in my day on the Human Genome Project, like it would take years to get a fungi sequence, and this thing basically occurred in a weekend, and it's at better quality than anything we could have ever dreamed of back then. But once we had that really good reference sequence, we wanted to get the chromosomes pinned down because 40 pieces isn't about right. It should be probably like 12 or 13 chromosomes. So this chromatin capture thing that Phage Genomics has is it basically takes the cells and hits it with a cross-linking agent. So all the proteins that are on the DNA kind of get glued to one another, and you can then sequence that material after you've kind of chopped it up. And the only thing that holds the DNA together is the protein. And so you can basically link pieces of sequence that are on the same chromosome this way, because the proteins don't cross-link to the different chromosomes, they cross-link to themselves. So you then bucket those 40 pieces. We bucketed it down to 13 chromosomes now. And so we've got the chromosomes all called correctly, and they're in NCBI now and annotated for anyone to use. But that's always the start, kind of the first gun, if you will. But after that, the right thing to do is then sequence 80 to 100 more of these fungi and see what the differences are between all the strains. Because if you can track the differences, you can start to associate some of those differences that might be related to whether they're making baeocystine or whether they're making psilocybin. So we did that. We sequenced about there's 84 public now. There's many more behind that are coming through that are various stages of assembly. Some of them, the ones that are outside the cubensis family, we haven't figured out how to get into Cilacida yet because they probably need their own reference genome. They're so different than cubensis. But yeah, if you go to Cilacida, you can probably pull it up now. It should be live.
Yeah, I was having an issue, but if you can pull it up, maybe you can pull it up there. I know that there's some. Well, sometimes when you launch a new website, it takes a minute to.
Yeah, it is as of Tuesday, but yeah, I can pull it up here. Let's see, you want me to share screen here?
Sure, that'd be great. Yeah, let me make sure I have it. There you go. Should be able to.
Bingo, there we go. All right.
There we go.
So it's Cilacida.net. So it's a lot like Cannapedia. It's just a strain registration system that's blockchain integrated, and this does a bit more than Cannapedia now. It's doing some microbiome analysis. It's also doing some work to do the assemblies. Each of these genomes are whole genome sequenced, and we assemble them out of the gate, which is kind of the gold standard. We don't do that on Cannabis per se unless people ask for it because it's more expensive. It's such a bit. These genomes are 42 megabases. Cannabis is like 890, so you can get about 20 of these done for the same amount of sequencing that you might use on a Cannabis strain. So there's not much point in like enriching for the 10 megabases you might care about because the genome's only 40 meg. So it's just sequence it all is kind of where we've been going with this. But let me see if we can pull one of these up. I think we were looking at this one today. It's Luminous Lucy's. So there's probably 4 or 5 different, I think at least 5 different spore providers that have contributed to this. Premium Spores, Sporeworks, Inoculate the World, and USA Mycology, and then I'm probably forgetting one of them, but you'll see them up in the DNA source category appear with links to their site. And these photos are theirs. We've just been taking the photos that they have on their site and replicating them here. But what comes out of this are all of the annotated variant files. That's what a variant call format is. It has every SNP and every indel finds the genome in just a long text list. Somewhat boring, but critical for really fingerprinting the strain. The raw sequence information anyone can take and upload and rehash this data if they have a different or more preferred way to do it. The ITS sequences are something that the fungal community loves to use. If they're out foraging and they find a new strain, they'll usually ITS amplify it and then Sanger sequence it. And that's probably the cheapest way. It might be 10 or 20 bucks to get something done that way. And it will tell you if you're cubensis or not cubensis. It doesn't have the resolution to really give you the strain name per se. Almost all the cubensis you'll find in here has the same ITS sequence. But when you get into tamponensis and cyanosins and azuricins, you'll see it starts to diverge quite a bit. And then these other files are binary after mapping files. This is after the reads are mapped to the genome. It gives you a binary format file that you can load into a genome browser. It's known as a BAM file. And a couple other versions of this that are annotated, where we have the genes actually layered in with where these variants are having. And then much like Cannapedia, we have the sequencing coverage across the pathway. So this is PSID, PSIH, PSIK, PSIM. These are the genes that are involved in modifying the L-tryptophan into various tryptamines. So this one here in particular puts the phosphate on. So this is important in dealing with psilocin versus psilocybin. And PSIM methylates. So this will change how many methyl groups are out there on the other end of the molecule. And that's often the difference between baeocystin, originacin, and the other compounds that are made with the plant. Now down below, we have a microbiome assessment and how many reads of this actually map to the genome. You want this number, usually above 75%, but this one's so 83% of the reads in this genome map to cubensis. The other 16% we then pour into a tool to look at what other microbes are there. The reason we're doing this is the cubensis growth is really dependent on, you don't want any bacteria around. It has to be pretty clean because the bacteria grow very, very quickly and the fungi grows very slowly. So if you get any bacterial contamination in there, it can take off. But we also just don't know which one of these are beneficial, right? These microbes you can't always view as being contaminated. Some of them might be necessary for rapid growth. So we're just trying to catalog that every time we shotgun something. And it might be different in spores than mycelia. So a lot of this work is getting done with spores. So the microbiome may be very limited inside of a spore isolation. But then, a lot of the assembly, now this is all getting sequenced with Illumina, which is a shorter read platform. So the assemblies don't end up in perfect chromosomes, but you can once you have a good reference built with PacBio, you can shortcut to Illumina and get a lot of work done for a lot less money. And these give you really, nice assemblies of each of these genomes. And this is another plot known as a blob plot here that's kind of looking for how many organisms are actually in the sequencing. You get these kind of blobs of reads of a particular GC flavor. That's a sign that, okay, there's another organism in here at low coverage. That's, this is around 10x coverage, this is around 100x coverage. That's probably contaminating the system a little bit. And this is just helpful to kind of keep an eye on what other microbes are present. But the funnest part is actually this system here. If you're a real nerd about this stuff, you can go and peek at all these variants. Let's see if I actually have the memory to pull up IGV here. I may not have that screen shared, but this is, you can go and look at every single sequencing read to see if the variants are supportable there. Hold on, I see I'm lagging there a little bit. You guys still hear me?
Absolutely. Yeah, no, this is fascinating. This is great.
Okay.
Man. I really appreciate it.
All right. So this should load up. Okay. Now, let me see if I can, you are sharing. All right. So let me share. I get the right. Let's do a new share. Let's do that one. All right. All right. So this is what's known as IGV. It's an integrative genome browser. It's made out of the Broad out of MIT. And what you're looking at here is every single sequencing read, and it marks it gray when it agrees, and then it paints it a slightly different color when there's either SNPs or sequencing errors. You can see these little things over here are like sequencing errors, but this is here a clear SNP. There's a variant there represented in about half the bases. This is about 116x coverage. That's really deep. We've kind of crushed this genome. You really only need to do this like 25 to 30x, but sometimes this is what happens. So we've got 47 reads that show it's an A and 65 that say it's a G. So that's an A gene mutation right there. And it's in a gene. I think we clicked on probably one of the PSI genes here, or one of the variants in the pathway. So now if we bounce back to the other sheet there, let me see, I'm gonna have to do a new share, get you over there. So anyone can do this as long as you have IGV downloaded for the Broad. It's open source software. But each of these variants you can go and peek at to see if, how good is the support? Because sometimes there's mistakes, it's like we call a variant and it's not really there when you dig it up. But this is really helpful for the public to kind of comb through because we got an email the day this went live from somebody saying, hey, there's something weird in one of your variants that the reads don't look like the support, but it's really important. Can you take a look at it? So I actually got that on my to-do list to do tonight. And then there's a distance calculator here. You'll see I'll pull up everything else that's very close to this. So the luminous Lucies are obviously coming up because we did a lot of these genomes in triplicate just to, we got them from multiple different providers and just to see that the names agreed in other providers. And surprisingly, actually in the cubensis space, it's pretty good. We were expecting this to be a mess, but I guess we had a different taste of this in the cannabis industry. But part of that is that these things are generally reproduced differently. They're reproduced asexually. So they can reproduce with meiotic events, but most people, I think, are sharing these spores that are probably fairly tightly related to the original fruit they came from. Whereas cannabis is often crossed, and so the names get confused with each cross. But you can also see the next nearest thing is this one, a renal volcano, which is one that came out of mushrooms.com. That was the other one I forgot. Similar plots down here, and you can see, it'll come back with the 3 luminous Lucies as being its closest, and then one of the penis envy strains comes out. What's really nice to see is that these samples from multiple different spore providers come back on top of each other, which is a good sign. It shows that there's actually some pretty good fidelity in amongst the spore providers. So on top of giving you nearest neighbors, it gives you the most distant ones as well, in case you want to do some crossing. And crossing is something we're still trying to figure out. Obviously, mycologists know how to do this, but it's not like cannabis. Cannabis has an XY chromosome system. And we believe cubensis is tetrapolar and that it has like four mating types, if you will. It might have 6. We're still trying to figure that out. Jason Slott found a bunch of pheromone B receptors that are interesting on chromosome 10. And Steve McLaughlin found 2 of the mating type genes on chromosome 1. And I think the combination of those things is beginning to give us some pictures as to which ones can cross with one another. So you can do, perhaps more successful breeding that way. And then we do the usual blockchain stamp down here for immutable records of the born on date of these things. And that may help in keeping things public or pursuing IP, depending on what people want to do with it. But our job here is just to document. So we always hash things onto blockchains. This one's probably on Dash. That's usually our default. And 9 days ago, a lot of these are actually up in NCBI back in October. So the earliest born on date is actually back then, but we just started stamping these a week before we put the site live. But that's the new tool. I don't think the phyllo tree is totally worked out yet. Oh, they did do some work on this. Okay, there's just something wrong with the color coding on the phyllo tree right now. It's not getting the distances color coded correctly. It's because it's keyed into cannabis right now, so we just have to adjust that, and the colors will come back again. But basically, these here are non-cubensis, which is why they're sitting off in this area. So this is Cilacida tampanensis. And I think this Georgia one, I've got to look up that one. I think that might be another Gallandois or Mexicana sample like this one. And there should be an Azuricens in here. Maybe that's the Azuricens, because there was a Oregon Coast one, which was an Azuricens one. So those are so different that the reads barely even map to the genomes. The mapping rates are down at like 10%, and it looks like there's no coverage over those genes, but they're in fact there. If you assemble the genome and look for them with protein searches, you can find them. So right now that's kind of the family tree of cubensis. Whole lot of samples over here that have some close relationships that we got to scrutinize, and I don't know, maybe it'll help in what we really need to do is chemotype these things, which we cannot do, but we'd love to collaborate with people who can. We don't have the expertise nor the jurisdiction to do that correctly. So the next chapter in this is to actually go and get as many tryptamines measured out of this as you can, and see if we can correlate those genotypes with the actual outcomes like we've done in cannabis. It worked out quite well in cannabis. We can now gauge just from the sequencing alone whether it's going to be a CBG producing plant, whether it's going to be cannabinoid null, whether it's going to be a CBD or THC or synthesize both. Now, we're never very good at nailing the exact quantity it's going to make, but we can certainly tell the direction the pathway is going to go based on the genetics. And I think the same thing is going to be true we're already seeing some variants in the pathway that are on our radar that might be playing a role in how productive it is making these things.
That was one of my questions, was that if you can tell the triterpene expression from the genomes yet, or if that's still
So Philippe Henry's done a little bit of work on that, actually. He just put out a little preprint yesterday for 420 for this. So I pointed to some of his work. He's gone through and really tortured what variants in the cannabis genome predict different terpene expressions. And I think he had some mention. He definitely had mono and sesquiterps in there. I think there was some mention of triterpenes and also the flavonoids. I think he was trying to poke at as well. So it's getting better. That's something that's a lot easier done in cannabis right now because there's so many labs that can measure those things that can also handle the DNA. I think the challenge we're having in the cubensis space is so new. A few people are really good at the sequencing, and only a few people in the world have licenses to even measure the compounds in the plant. But that's changing, and it's looking like Oregon's going to open up on this, and a couple jurisdictions here in Mass opened up. Cambridge, Somerville, Northampton, all decriminalized at least. I think Berkeley and Oakland did as well. Denver. But Canada, I think, is probably the ground where a lot of that's going to happen. So we're speaking to some folks up there to see if we can get some chemotyped material DNA extracted for us, and we'll sequence it. And if they can get the RNA, it'll be even better. We'd sequence that as well, and that'll help decorate these pathways a little bit more.
Yeah, definitely looking forward to that. For sure, it's definitely an interesting project that you're working on. The one other question I had for you, particularly in regards to this set of data, was have you sequenced PES Amazonian from SporeWorks? Was that one?
I think we have. That name does ring a bell. And there's Oregon Coast. Let me see where. Like, there's got to be.
That was definitely a good yielder for me back in the day, and a good reliable cultivar.
Right there.
Nice. Yep, that's it. Very cool. I'll have to dig into that later.
Yeah. So that one's there. The thing that was hardest about this project is that they all have very different spores, and that are they range from whether probably about 80% of the samples cruise through with our prep. 20% of them we had to fight like hell to try and get DNA out of the spores, and we don't know why. And we've since figured out how to hammer the spores and break them open more, but that was not that's what led to a lot of the replicas that you're seeing in here, is that we kept trying them over and over again and trying to get more and more DNA out of them. So the best way to do this now is to send a spore print or a spore syringe, and we can get DNA out of those and sequence the entire genome. Now, there are some subtle drawbacks to sequencing spores, is that you don't get a haploid representation of the genome. So the mating type loci have variants in the genome when you look at them. If you were to sequence this from like a clonal colony, you might see a slightly different picture in the mating type regions, because it would be one mating type, whereas the spores are a collection of all of them. That's the only thing we've noticed so far difference between the two. But the rest of the genome seems like it's readily callable without any problems.
Very cool. Oh man, I lost my train of thought on that follow-up question there.
You have some more variants in this one.
Yeah, it's one of my favorites. You can stabilize it out to about 1 gram per, and it was a good producer for me, at least, and strong.
Looks like it's closely related to Golden Teacher,
huh? I didn't know that. And Golden Teacher is related to PE, isn't it? Or no?
Oh, I would imagine the PE isn't the PES one related. I thought that would be related to penis number. Yeah.
Yeah, I thought they were close.
Let me see what we got here. These are all coming in really tight. We probably have to expand that a little bit and see if we can pick up any of the others.
Have you learned much more about the MAOIs and the genomic expression on that? Is it universal, or is it hyper specific between cultivar types? Or
we haven't done any of that work. I've just mostly just been bookworming that out of the literature, and there is some work suggesting it's mostly expressed in the mycelium. So That's I'm kind of wondering what will happen in the mushrooms that have sclerotia, because that's kind of some kind of state that looks to be like a mixture of mycelium and fruit, and maybe those have higher beta carbolines in them. But no, we don't have the capacity. No, those compounds I bet would be different. That's a good question, is whether the DEA would have any issues. One, it's not our expertise to monitor those things, and so we wouldn't be doing a very good job at it. But two, I'm always questioning like whether it's legal in Massachusetts, and the carbolines might be. I'd have to look into that, but we don't have a license to express any compound. Now, I have heard from some of the spore providers. I think it was Drew at Inoculate the World mentioned he had some of his cultures tested to see if there was any psilocybin detected in the culture state, and he doesn't have any detected in his. So that kind of, I think it's got everyone wondering what does this mean based on what the DEA recently did with type 1 cannabis seeds, because the premise there wasn't about viability, it was about compound percentages. And so if it's undetectable below some limit, then the cultures might be able to go in the mail too. And that means we could probably do RNA expression analysis off of cultures, which would be very interesting. Because I'm sure I don't think we're going to get much RNA out of the spores, and there's not much gene activity going on in there. But once you start growing these things, all the different genes should turn on in the spawning process, which might be important to document.
Well, I definitely am excited to learn more about this and see how it grows in the future as you get more and more samples. And there's certainly no one else doing this in a public fashion like you are. And I really appreciate all the hard work you're doing down this space, because it's going to be really critical to preventing some of the different patent wars later on and other crazy bullshit that goes on.
Yeah, I mean, there is a vibrant community that is doing fungal sequencing of ITS, where they amplify the ITS region. You can do that in the field with these things here. This here is like you probably can't see this because I'm sharing a screen, right? Maybe I'll unshare. Yep. There we go.
There you go. Now we can see it.
But yeah, these little devices people can take. You can't see it now. Let me hold it right there by my face. These are field portable PCR units, and so people are using these to amplify the ITS region. And then you can send that in for Sanger sequencing and get at least an understanding of whether it's cubensis or non-cubensis or Cilacida, tamponensis or something. So there's actually a great community doing that, and a lot of that stuff's going public, and we're trying to integrate that into Cilacida. And then a couple other academics out there are putting some genomes public as well. So I think a paper just came out with about 90 some odd ITS sequences that aren't just Cilacida. They're like a whole portfolio of mushrooms that were in a museum that they weren't certain how many of them were hallucinogenic and how many weren't, so they wouldn't catalog them all. And so the data is starting to build. There's starting to be a nice public repository, and this is certainly one of the larger deposits ever put public. It is fairly focused on cubensis. It doesn't have a whole lot of activity outside of that. I mean, we have some of those genomes sequenced. We just haven't figured out how to integrate them to this viewer because they're so diverged from cubensis that the data just kind of looks kind of meaningless at the moment. So we have to build some other reference genomes and have fork this so that Cilacida can then also handle cyanosins and all the other ones that people are interested in. I think right now some of the laws, however, are constricting the use of those. I think Oregon is only legalizing cubensis.
Oklahoma is actually damn close. I think just the governor has to sign off on it.
Oh, really?
Legalized research. Yeah, they have two psilocybin mushroom bills, and they have two more that are about to be introduced that haven't been introduced yet.
What do you need to get a research license down there?
Yeah, they're aiming to commercialize it and be in line with Oregon having testing, ultimately having the testing, cultivation, and dispensaries. But we'll see if it goes through or not.
Oh, that'd be huge. That'd be very helpful. Yeah, keep me apprised of that, because we'll. If there's anyone down there that needs, wants to learn more about this, if the regulators are concerned. I mean, some of the regulators throw up concerns saying, "Oh, we don't have a way to identify this from toxic mushrooms," and we do now. Like, we've got enough sequencing on this that we can differentiate cubensis from all of the other concerns that are out there. That they probably don't know that. and they'll probably bring up that that foot fault, being like, "Nah, it's too dangerous. You're going to mix up these mushrooms." I think that's addressed. The capacity to measure any microbe they care about in this, I think we have addressed as well, because we have protocols that can do PCR out of these out of spores, and we have tested those on DNA extracts from fruits. So if they're concerned about any other microbial contaminant, they usually are concerned about any edible E. coli and Salmonella. We've not seen the evidence of those microbes in this yet, but that's usually what they kind of carte blanche put on any edible, even in the cannabis industry. So those shouldn't be too difficult to capture out of this. And if there's any other microbe that tends, I haven't seen a lot in the literature of all the toxicology I've seen the literature on this is usually from mistaken Galerina marginata, or I don't even know how people mistake the amanitas for this, because they're usually red, but there have been some amanita poisonings, probably because people were confusing them with a different amanita that they're looking for. But there's reports of like liver failure and kidney. It's not a good way to go. I mean, if you get it leads to like liver transplants if you ever make this mistake. So it's something you don't want to get wrong. But I don't know that there's a high number of them in the literature. It seems to be a really rare event.
Yeah, it's certainly really exciting to learn about all these new things and down that space for sure. Definitely looking forward to that. What else is new in the world of genetics testing and things like that in terms of cannabis? I know you've been doing a ton of work down that space as well, and you got every time I'm on your website, you have a new test on your. Let me pull it up here.
Yeah. So we have actually 14 tests for viruses now, but we've only released probably 5 of them because we're waiting for molecular confirmation that these things actually exist in the plant. So what do I mean by that? Well, hop latent viroid, people have sequenced that genome out of the plant. They've proven it actually exists in cannabis. So that one gets used quite a bit. That's probably the most popular of the virus tests that are out there. Lettuce chlorosis virus, we see more in Israel and in the East Coast. That one gets used a bit out here. Beet curly top is showing up quite a bit in Colorado. It sounds like you've had it as well down in Oklahoma. Yep. And then tobacco streak was just found in Colorado. And that one, we had a test ready to go, but after seeing that paper, we're modifying it to make sure it can capture what they found in Colorado because it was quite diverged. And we just put out a tobacco mosaic one, but I'll say the tobacco mosaic one, that is one where we got begged to put it public, even though we have not found it in cannabis yet. So I've heard rumors of someone finding this in cannabis, and so we're just cross-checking our assay to make sure it actually hits what they found.
People always anecdotally say a friend of a friend of a friend has tobacco mosaic virus, but no one can ever display it, right? Yeah, exactly.
And I think a legitimate pathologist has now found it, sequenced it, and it's different enough. And I'm hoping they call it something else. Cannabis mosaic virus. It probably has probably earned itself its own name, but we'll see. But that we haven't run into that quite a bit, and maybe it's because there's just not a lot of testing going on for it. But that one seems to be what everyone blames it on that, even though I've seen HLV plants produce a mosaic pattern on leaves too. It doesn't happen all the time, but some of the ones we worked on with Colin out here were like, we first screened his plants against all 14 because we figured, oh, this looks like a mosaic virus. We got to hit all these other mosaics that we have. And they were all negative. It was surprising to us to find it was hop latent viroid that was doing it. But yeah, that's been an interesting project because we went through and tested these were heirloom plants that he did not want to throw away. I mean, he's had them for 20 years or more. And so some of the really old genetics that go back to the original Sour D, some of the original ChemDog, some of the original, a lot of early lines. So, just one test and throwing them out was not an option. We tested the samples like in triplicate, like 3 times a week for a couple of weeks to make sure that they were all, in fact, consistently showing viroid levels, and all the internal controls were working correctly. And then anything that was a really like late CT, we were sure to repeat multiple times just to make sure we're not missing this. But we also went through his plants and looked at them in multiple tissues, flowers, stems, petioles, roots. It's everywhere. It's in the roots. That has me thinking this may transfer plant to plant through the mycelium, because there is some documentation of viroids doing that. So its transmission is all believed to be mechanical, but we've not yet caught insects in the act of moving it. I suspect that happens. We've not really interrogated the mycelium very much, but there may be some, if you have plants on two different edges of a root bed, that's possible that it could transfer that way.
The most interesting observation I made with Mosaic Expression and cannabis was, I was at a grow. I'm going to leave the grow nameless, but I'm sure they would prefer I did so. But I was at a grow and they were running a test between 4 different lights, and they had the same size grow room with the same cuts in each room running a side by side. And in one of the rooms, the difference in light spectrum caused one of the strains to hyper express the Mosaic virus. And the only difference was the lighting. And I know that for a fact because they were running this test to see which light they were going to use on their larger facility there under construction. So I thought that was a really strange observation to see that the light was having an immense expression on effect on the expression of the viroid, specifically whichever type of mosaic this was. Kevin, I can talk to you about this offline and maybe even get you some tissue samples off those moms. But because I think they still have moms down there from those moms, but it was only under one of the lights that it expressed it, but it was the same cut under all 4, but only one of them showed it.
Was it more, do you know if it's more red or blue light that was doing it?
I'd have to go back and look at the photos. I have photos of it though.
Oh, wow. That's really cool. No, I'd love to catch up with you on that. Cause that's, it's really it's annoying in that these are latent viruses, right? So I think Colin, he had about a dozen plants, 8 of them were positive, 2 of them showed symptoms. Right. So that was a bit surprising. And now after he knew they were positive, he went back hunting for very specific phenotypes, like, are there any brittle stems or what have you? And he started to say, okay, yeah, these guys aren't moving along as quickly as I hoped they would be. And their stems seem brittle. So I could see how they're kind of asymptomatic or partially symptomatic, if you will. But that's, there's a lot we learned in the last 2 years about dealing with PCR with asymptomatic viruses. You got to be really careful. I mean, it's one thing to have clones and just, they're disposable, right? So maybe any PCR signal you see, chuck them. But when you start dealing with some of these heirloom genetics, I would not want to be throwing out mothers if you had a single PCR test that was out in the late 30s, because it could be a false positive, or it could be a plant that's resistant. Like people who are resistant to COVID are still PCR positive, right? Your immune system keeps it at bay, so you don't have high viral load, but it's not zero. So I don't want us to get in this habit of just clear cutting all of these heirloom genetics off of late CT single tests. When if it's a really important mother plant, I think you might want to keep it and study it, because those viroid loads, they're like 1 million-fold lower, right? That's significant. That's something to keep an eye on. Like most of these viroid loads, when they come out, they're screaming hot in every tissue. It's not like it's black and white. Like they're really hot at CTs of 20. So when we start seeing stuff that's really late, we think false positives, contamination, or maybe it's biologically very interesting and you should keep it. It might be one that you want to breed with because it happens to keep the viroid load very low, and maybe it's never affected by it. Maybe it's a contamination risk and you got to be careful. But at the same time, if it's 1 million-fold lower, the contamination risk is probably 1 million-fold lower as well. It might be an interesting one to keep, breed, or maybe tissue culture your way out of it. There have been some people reporting that cold tissue culture seems to slow down the replication of this thing while the plant can then clear it.
Does it work the same way that it does in some other organisms as far as reference genomes, where maybe the plants are storing that genome as a reference material for antibodies or anything like that, or does it not work that way in plants?
So this is a most fascinating molecule. I've got to say it will blow your mind trying to think of the chicken and the egg that came up with this thing, right? Like if you want to be an advocate for intelligent design, this would be something to study, right? Because, all right, so this is a molecule. It has no viral code on it, right? It's just a 256 base pair RNA molecule that's circular. and it happens to be a ribozyme. All right. So for those who aren't familiar with ribozymes, they believe the earliest forms of life were ribozymes, that they were RNA molecules that could fold in such a way like an enzyme that they're catalytic, that they actually perform reactions with other metal cofactors, maybe magnesium or what have you, and they can actually cut other pieces of RNA. So this particular ribozyme, if you look at it, it has a couple bubbles in it. It's not a perfect stretch of double-stranded RNA. It's circular with a lot of bubbles. And those bubbles, we believe, are there to evade a lot of the RNases that typically clear these things. There's a lot of endogenous RNases in plants that go and chop up double-stranded RNA because it looks like an invader. It's also involved in a lot of the Argonaut dicer pathway is involved in this, right? So this thing has figured out a way to get into a plant cell. It doesn't code for any genes. It's just a ribozyme that can cut RNA, and it's targeting itself. So this circular RNA, when it starts to copy, it's called rolling circle amplification. If you can imagine, one primer gets on that thing and it's just a gerbil wheel that goes on forever. And so you get this long RNA molecule that's made off of the original circle. And then the ribozyme, the double-stranded versions that are still floating around, go and cut that into 256 base pair pieces of little RNA. And the plant genome or cell somehow circularizes that with a ligase and puts it back into a circle, and the process repeats itself. So somehow this molecule's figured out how to hijack the replication in the plant, cut itself, and make more of itself without there being any gene involved. It's totally fascinating. They do seem to be a little bit RNase resistant as well due to the feature of these bubbles. We've tried all different types of RNases on these things to clear them, and they do have differential activity on it. There are some RNase genes in the cannabis genome as well that we're looking at. There's one known as RNase 3 that may be playing a role. Like, why did Colin have 4 plants that didn't get it? Where we sequenced all those genomes and probably years before we figure it all out. But we're hoping there'll be some signatures in some of these plants that are less susceptible to it and keep lower viroid loads. We may not be able to get rid of it 100%. Like this concept of zero COVID didn't really work. I don't know that zero HLV will work either. We may have to find plants that just keep the load so low that it's not symptomatic and doesn't affect yield. Certainly there's people disagree on that topic. The nurseries just want to go zero because they have to. They're in the business of cutting and moving these things around. But what I'm most worried about is clear cutting so much of the ancient genetics that are around if we deploy hyper aggressive PCR everywhere. Particularly if this is, there's a lot of artifacts that can happen just with statistics alone. If you turn this test into, like, let's say a non-quantitative test where you don't get this information about CT values and you pick some places positive and negative on that scale, you run into a lot of problems when it's at low population frequency, because all tests have a false positive rate. And even if it's 1%, that's close. If that's close to the disease prevalence, you're going to find false positives as frequently as you find positives. So there's a lot of Bayesian statistics. You gotta be very. I'll point people to a Substack on this that I wrote, or a blog that we wrote about this. But when you get involved in presence absence testing, you got to be very careful with Bayesian statistics to make sure that you don't end up having your false positives just eat up your entire statistical significance. But that was happening in COVID testing for quite a bit with people who the earlier tests didn't have the right controls, and there's a lot of this crap that went on. But what I don't want to see happen is this thing rages through California and we just slash and burn all the genetics out there, not paying close attention to there could be some out there that consistently have lower viroid loads, and we want to capture those and see if we can breed with them. The other thing I think that needs to be better studied is what percent of the seeds carry this, because a lot of if you look to the genetics of the seeds, a lot of these RNases get hyperexpressed in the seeds. And they believe that is meant to purify the seed from not having any viruses in the next generation. And so I've heard rumors, although I've not seen a study on this, I've heard rumors from various people in the field that there might be a 5% contamination rate in the seeds, but like 95% of them are usually clean of it. So, selfing these things and making seeds is another way to potentially purify them outside of tissue culture.
How prevalent is it in quote unquote landrace cultivars? Do we know yet?
I don't know yet. No, I mean, that's probably a better question for some of the nurseries that have been handled a lot more diverse lines than we have. We're about one step removed from this because we sell the kits to a lot of the labs who then work directly with a lot of the growers. So we only get kind of aggregate information as to what the positivity might be in various areas, but we don't have any understanding of who has it where and what plants are getting it.
I was just going to say, my follow-up question was, is there any populations that you've seen it explicitly less dominant or less prevalent in?
Not that I would call statistically significant yet. I mean, there's the study we did with Colin, and that was just 12 samples, and 4 of them didn't get it. And I don't know if that's just he had them isolated enough from the other ones, or if there was something about those that significantly thwarted it off. I mean, the right thing to do would be to probably clone those things and then put them into the same territory and maybe even mechanically introduce the viroid to those to see if they harbor to the same degree.
So another question I wanted to bring up, just because I've had a couple people bring it up a lot recently about herming. Can you talk to us about the genetics around hermaphrodites and hermaphroditic traits in cannabis and kind of what causes that? I've heard that too much back crossing can cause it, and a whole bunch of other kooky ideas as to what's going on. And I know that you're kind of an authority on this type of stuff, so I figured you'd be a great person to ask.
I wish I had a better answer other than we hope to find out, because so what we built into Cannapedia is a Y chromosome coverage map. So if you go in there, you'll see when we whole genome sequence something, we just count the number of reads that fall on the Y chromosome and how many they call on the X chromosome. Right now, we use the Jamaican lion reference is the only one out there right now that has a Y chromosome. So, and it's a big chromosome. It's like 100 megs. So that is that counts the density of the reads. There may be some variants that exist on the Y or on the X. We also have the X identified, so we can look at if there's different variants on the X and Y right now. That's about as far as we've gone. We haven't sequenced this RNA yet. It's killing me that I haven't done it yet, but we did collect RNA on plants going through the transition to see if we could see what genes were lighting up in the process of hermaphrodization. And we did it both directions. We did it with ethifon going one direction, and we did it with silver thiosulfate going the other direction. But we just haven't gotten that stuff onto PacBios yet. It's been sitting in a freezer for way too long. But that might decorate what parts of the gene and the genomes are responsible for this. And it probably needs to be done, as you mentioned, across a lot of different cultivars, because I have also heard the same thing, that some of the lines are train wrecks. They just hermy really, really readily. And that's probably genetic from what we know in hemp. John McKay's done some good work on this, where they were collecting hemp samples that had different monacity rates. Like, they would be, almost permahermes, if you will. And they'd have male and female flowers, but they'd have them at different ratios. They consistently have them like at 90-10 or 50-50. And I know they've been doing some work digging into that. I just don't know what's come of it. But that might in fact inform on if there are any genetic components that predict that.
I wanted to mention too, so the only time I've ever got my hands on Jamaican lion was at the Regen conference out in Humboldt. A guy was giving out clones. We brought one back.
Cool.
To
is this Jeremy by chance?
What was that?
Was his name Jeremy by chance?
I'll be honest with you, I don't remember.
Yeah, all right.
But it was at the first Humboldt Regen conference when it was at the elementary school up there. And the weird trait that I found with that, because we caught a couple clones off of it and flushed it out, a great plant in terms of yielder, but if we kept it under 24 hours, she would throw a small amount of male flowers. If we put her under 18 hours, she wouldn't throw any anymore, and she'd act like a normal plant. But the only time we could get her to throw any herm traits was under 24 hours. So there was some type of trigger. I don't think I've ever put it under.
Yeah, that's an important point. I don't think I've ever done that to it before. Now, the genetics I got were seeds out of where was I? I was in Hopland. There's a dispensary up in Hopland that Martin Lee brought me up to. I'm forgetting the name of it. But they were up in there. They had seeds up there, and that's where I picked them up, and eventually got in touch with I think it's Ganja Genetics. And they shared some history of that. It's apparently been around for a decade or more. It was, I think it won a Cannabis Cup, a San Francisco Cannabis Cup back in like 2013 maybe, or It's a while
Yeah.
ago, but
Yep.
yeah, we had good luck with that. And that we had a hard time getting that thing to take any powdery mildew. I had a powdery mildew loaded plant, just like snuggling with that thing for months, and it wouldn't pick it up. I don't know how well that's. We're a couple descendants, I think, from what actually won the cup in 2013. I think we may be 3 or 4 generations down from that.
Random observation, and maybe this will also cue into some of the genomes that pick up on silica, but we did a ton of different side-by-sides with PM and tomatoes, and PM with a couple of cucumbers and a couple of other vegetable crops at Aquaponic source, because that's what we could legally do in the space. We had a separate offsite cannabis spot, but silica levels played a huge role in the PM resistance and the genome expression. I know that there's research on corn and wheat on the genome expression with minimum silica levels. So if it's below a certain threshold, they don't activate that gene that increases that mold resistance. We've seen that directly, and I could give you exact parts per million thresholds in aquaponics.
Now, were you seeing it jump from tomatoes to cannabis? Because there have been 1 or 2 more powdery mildews found on cannabis. I think someone's now confirmed that.
I can speak on that firsthand. I can tell you that the shit that I dealt with in California was way different than the stuff I dealt with in Africa and Zimbabwe, and different than the stuff I dealt with in Jamaica, and different than the strain I deal with in Oklahoma.
Yeah.
The one in Oklahoma responds much better to Bacillus pumilus, whereas the other ones respond really well to Bacillus subtilis. But the Oklahoma one, I can spray it with cease and it'll knock it back, but doesn't get rid of it. But if I spray it with something like Sonata, which is a Bacillus pumilus, or what's the General Hydro has one called HydroGuard, or someone in chat might know. General Hydroponics has an equivalent that's a Bacillus pumilus concentrate. Both of those work really well in Oklahoma, whereas the Bacillus subtilis doesn't really do all that well. So there's definitely a difference in species for sure.
So there has, I know of, I think about 3 different genomes so far that have been documented. One is Golovinomyces, I think it's Ambrosia. One is Golovinomyces chicaracearum. There's a third Golovinomyces I think I saw recently reported, and then I think there's even Potosphaera, I think, from hops they think has jumped as well. So I wouldn't be surprised if there's like a dozen of these ones. And we've had a test out there that only really targets one, because it's all we knew about back when we sequenced a powdery mildew genome many years ago to try, and it was a disaster because it was very hard to sequence. It's very repetitive, so we didn't really get the whole genome in perfect shape, but we got enough to get a PCR marker off of it. But we're probably missing some with that in if there's different flavors of it now being discovered. Fortunately, the Golovinomyces tend to all amplify. I shouldn't say all of them, but the ones so far that have been discovered, I think, are working fine with the primers. But we have a different set now for Potosphaera. It's just we don't know which jurisdictions need these and whether all the tests have to have all the different powdery mildews on them. And it's not something people have been really eager to qPCR test for, mainly because they usually can detect it by eye without any, and there's usually an easy way to deal with it, like you mentioned, or some people are using hydrogen peroxide sprays and other things to just knock it back. And there are ways to breed your way out of this. These things don't grow on any of the total yeast and mold plates. So it's not a yeast and mold that's going to make your sample fail in any cannabis testing lab. And so as a result, a lot of folks just don't care to necessarily track it. But for research purposes, a lot of people are, they're buying them and using it just to figure out what they have. But I wouldn't be surprised if in the coming years people find more of these. I think what we learned from sequencing these psilocybe spores, we want to go back and apply to the powdery mildew, because one of the problems we had was lysing open those things. Cracking them open to get enough DNA to do a genome out of wasn't easy. We'd always end up getting all the other microbes but it, because when it's there, you tend to see like the penicilliums burst up, a bunch of other things break out. Plant's immune system's like wrecked, and so all these other things are there as well. And they lyse open more readily, and you tend to sequence everything but powdery mildew when you go after it. So they do need a real. We've been using these enzymes. Actually, an enzyme we cloned out of cannabis to fight this, to put in our kits. So our yeast and mold kits now have a thaumatin-like protein in them that you do an enzymatic digestion to help crack open some of these molds. Some of these molds build these really thick cell walls that are loaded with glucan and chitin. And so to cut that open, we throw enzymes at it so we can ensure we're getting all the DNA in a given test. But that was also critical in cracking open the cubensis spores. We've been throwing that in there to basically digest the things overnight, and we end up getting much more DNA out of the preps when we do that. So, oddly enough, some of the powdery mildew resistance genetics has kind of pointed us into a direction on how to better analyze fungal genomes, which is kind of an unexpected twist.
That's really cool. I never would have thought of it in that in that way. That's really neat. What other questions do we have here from chat here? 1 second here.
Oh, can we sequence humans? I think I see in the chat.
Yeah.
So I used to run a CLIA lab that would sequence humans, and it's so damn regulated that we don't do it anymore. Sorry. You have to have a CLIA lab to do it. And the FDA likes to get involved when you start making clinical calls on human genetic data. HIPAA law comes into play, as it should come into play. But that's a very, very tightly regulated space now to sequence humans medically. You got to have a physician sign off on every test. There's probably some research use only places you can do it, like 23andMe, but there's a limited number of things that they can tell you based on the overhang of the FDA.
So is there any other pest-related genes that that you've also discovered, or any other work that you've done aside from PM down that space?
No, and on that front, we've mainly been focusing on a lot of the viruses and less of the. We have the mold resistance genes on a chip now. So yeah, I guess this has been a while since we spoke to you. Probably haven't spoken to you about this, but. All right, so two things in that area are, there is a SNP chip now that has 85,000 markers on it that Eurofins manages in Europe, and we manage in the United States. And this has a lot of the genetics we basically tiled the chitinases, the TLPs, and then a lot of the genes that had MLO in their. MLO is a mildew susceptibility loci that you'll find. David Jolly actually did a really good paper on that stuff. And so we've tiled a lot of those regions on that chip. I think there's like 80 markers across. There was a PM1 marker that came out, I think, from Dewey Scientific, I think, published that. And we already had that region on the chip. That was on our radar. Just having the annotated references, we knew that one was probably going to be involved. But that one's hyper tiled as well. So there's other tools out there. The second tool that we put public was when we do cannabis sequencing. If people don't want to sequence the whole genome, we have this SureSelect system that enriches the coding regions of the genome, about 10 to 18 megabases of coding region that we pull out of the genome. The entire coding content of the cannabis genome is probably like 42, 43 megabases. So we're not capturing everything, because when you start capturing everything, it makes more sense to just sequence the whole genome. So we have something that captures about a quarter of the coding content. It hits a lot of the cannabinoid genes, the terpene genes, the genes in the pathogen response. That used to be a panel that we use internally, and we decided to put that public so anybody can order that from Agilent, send it to their local university, and get stuff sequenced without us if they want to. And that's just part of keeping it neutral, if you will. Like, open source like that means people can check our work, and it builds a little bit more trust. Other people can sequence the same panel that we're sequencing. If they need us to analyze the data, we can pump that into Cannapedia, kind of turn it into a SaaS model, if you will. But that community design you can now order, and I think that's been helpful for the whole Humboldt legacy project out there, because they now can point to hey, you don't have to necessarily send it to Medicinal Genomics if you're concerned about their private affairs or their business motives. You can buy the kit, send it to a university, get them to sequence it, and send you the data. And if you need help with the data, there's universities can either help you or we can help you. But it's just meant to keep some more of these tools out in the public space so that more of these questions get answered. I think the biggest problem we have in cannabis genomics is that there's so much damn data that it's somewhat foolish to hoard it. putting it public means other people see stuff you don't find, and that's what's happening. Is we're seeing people now publish multiple different markers that are pointing to new genes that are involved in these things. So if anything's new, it's not necessarily new gene discovery. It's more tools have been thrown out there for other people to poke at the problem.
How is the mapping of terpene expression in cannabis going? I know we touched on that a little bit earlier with the mushrooms with the triterpenes. What do we know so far on the cannabis side?
Oh, I thought you were asking about that on the cannabis side. I'm sorry, I answered that wrong. But the most recent thing I saw was from Sean Miles' group, I think, put some work out, and then Philippe Henry put some work out where they're tracking various SNPs that are predicting the classes of terpene expression, and they have some interesting papers. So I would Keith Allen's done some work on this as well. I haven't seen his most recent work, but those are probably three of the names to go after to dig more into that. We're combing through those papers to see if we need to port any of those tools into Cannapedia, if they're predictive enough for us to start calling things off of the markers that they've seen. We've done some of our own PCAs on there, where we don't have the same diverse chemotypes in Cannapedia. So they've really gone out and looked for ones that have unique flavonoids and have unique some other extreme terpene profiles that we were missing. I think a lot of our genetics are really heavy in beta-caryophyllene, terpinolene, and limonene and pinene, but we were missing some of the ones that are a little bit more sesquiterpene dominated.
Someone else in chat said they are super loving your PathoSeq. Do you want to touch on this for a minute?
Is this the Aspergillus kit?
Yep.
Yeah. So that one just got through AOAC approval. Our South Stack got through AOAC as well. So AOAC is now in place to basically get a lot of these kits harmonized in the field. A lot of different players out there all using different tools. And so there's now a process in place where you run these things through to make sure they meet certain standards. And a lot of that's been enabled because there are now standards. For the longest time, we haven't had standards that we could ship across state lines. But these are all the different organisms that we can scan off of a qPCR machine. You can either use a BioRad or an Agilent urea. We tend to be on the Agilent side of this more because they've just been much easier to work with. And we've just got a good rapport with them. But they both give similar CT scores with these kits. We validate everything on both platforms. But the Aspergillus one is probably the one that we've poured the most heart and soul into because it actually picks up 5 things in one well with 5 different colors, which is a bit tricky to do in PCR because the number of wavelengths that you can monitor simultaneously. But that one gets Aspergillus niger, flavus, fumigatus, and Interius. And then there's the 5th color is internal control. We always have a cannabis internal control to make sure that you extracted actual cannabis and that we can monitor how much we have relative to that. But this one's been eaten up quite a bit in California and other places because it's fast, easy, and a quick way to get to Aspergillus levels. These things are kind of tricky to culture. There is another paper we just put. It's in review right now, but it's out as a preprint that really came out of some work we're doing in Pennsylvania on the enterobacteria side. We kept having people call us. We were getting actually 2 different answers in Pennsylvania, which is weird. One group was saying we're overcalling enteros, and other was saying we're undercalling it. And this just to us was like, wait, it's doing one or the other. It can't be doing both. So what the hell's going on? And so we got a lot of these microbes in and then started going through a lot of the different enterobacterias in ATCC and biobanks and then plating them. And what we discovered is they plate almost in a binary fashion. They don't plate at 36 and they do show up at 26 and 30 degrees Celsius. So the temperature dependence on some of these enterobacteria is extreme. And if you're not careful and you're growing everything at 36, you'll find none. And someone else will grow it at 30 and find a boatload. And so we've kind of documented that and put all that work public just to kind of warn the world. There's a couple of these enteros that can cause nasty gastroenteritis. Are on the CDC's list for bugs to be concerned about, but they you can easily not find them by growing at too hot of a temperature in the culturing conditions. The PCR on the sequencing side picks them up. It's temperature independent, so it finds them regardless. But This is what often gets thrown at us with PCR is that, oh, you've got a live dead problem. You're picking up all this dead DNA because I don't get anything growing. Well, it depends on what you grow it on and what temperature you grow it on. It's probably there. It's just not growing under the conditions that you selected to try to pull them off the plant. So there's a whole little story on I don't know. I you can see from the background I have them here. I kind of dig on microbes. So these stories are always really fascinating to me. The same thing might be happening with the yeast and molds. We did a yeast and mold study out of Michigan where we looked at multiple different medias. Like this one behind me is DRBC. It makes really beautiful images, but it's got 3 different selections on it. So not all the yeast and mold grow on it. We tend to get an order of magnitude less mold off of these plates than we get off of PDA. The problem with PDA is a lot of bacteria comes through on it. So a lot of people just use PDA with chloramphenicol and most of the bacteria goes away, but some of the yeast goes away with it. So we've done a study looking at different media types, and those can each vary by an order of magnitude on your CFUs. We haven't done is temperature. We don't want to do temperature next, and we want to do cannabinoids next because we think the cannabinoids that come off the plant may be playing a role as an antibiotic, and what microbes can and can't grow based on it being a type 1 or a type 3 plant. The terps probably play a role too. The terpenes are known to have, if you actually fumigate plants with antibiotics, the terpene profiles change, which is kind of just some fascinating papers out there showing that there's some kind of communication going on there.
That makes it makes a ton of sense. I mean, anyone that understands how to maximize terpenes, how do you maximize terpenes? You maximize the amount of beneficial microbes that the plant's exposed to in its foliar system and its root system, right? So that activates a genomic expression in the plant's immune system, and it creates secondary metabolites that we call terpenes, flavonoids, et cetera, et cetera.
Yeah.
So that makes absolute sense. And I think that's also why, there was a post this week about Kangen water. I think that's another reason why you should not use Kangen water. Why do you want to sterilize your plants? It's kind of it goes against what you're trying to do, especially if you're spraying with probiotics and you're trying to stay organic.
Yeah, I mean, this is something that drives us nuts because a lot of the states are rubber stamping these total tests in place. And I mean, we have them and we spend a ton of time trying to calibrate those things to plates, which is really hard to do given all the variations I just mentioned to you. But we worry that they trap growers into this boy in a bubble type of grow environment, which is not where they want to be. And it's probably not a very long-term viable solution to try and have a hyper-sterile growing environments. I think so the total tests worry us because they're not measuring anything clinically meaningful. They're measuring a microbiome, most of which is probably beneficial to you and the plant. So we've been trying to encourage folks to look at the species specific approach here because you just want to, the ones that are toxic to human, get rid of them, right? That makes sense. But this idea of like, if you're above 100,000 CFUs of total aerobic bacteria, it's like, that's not a clinically meaningful number. It's just it's a tarot card that they're kind of it's not actionable, right? And it may in fact induce a lot of bad behavior by having people now that, they're going to take the plant and start treating it with ozone and all types of other things to try and kill this. And then the ozone has side products you don't know about, maybe on the terpenes, it may form formaldehyde, and it's just a big chain reaction of unintended consequences when you just superimpose the stuff. So we've really been trying to get the word out, like just move towards species-specific testing. You just go after the pathogens you care about and not play this games on counting total aerobic and total yeast and mold. It shoots 2 of our tests in the feet, but those 2 tests have the highest amount of like service calls because they're always in disagreement depending on how you plate. You use a different media, you get a different answer, and that's the problem that we've inherited. Unfortunately, the gold standard out there isn't really locked down. So you got to calibrate to something on one media type. And the moment someone deviates from that, they're going to get different microbial numbers. So, the total tests I think are, they're not necessarily actionable, clinically meaningful, and they trap a lot of cannabis that probably is just fine for consumption. And I'm afraid it's going to be a genetic bottleneck, that there'll be certain plants that just can't get through and survive with lower loads. And so you're going to lose certain genetics from the field because they don't fit the paradigm of growing under these regulations.
Absolutely. We run into the same thing with aquaponics with non-human pathogenic E. coli. There's a many species of E. coli. Most of them are, don't affect, livestock or humans. A
Right.
small percentage of them do. So, in fact, many species of E. coli are used to manufacture many of the pharmaceuticals that, people listening to this show actually
Absolutely. Yeah.
consume every day. Just so that you guys are aware of how neutered some of these have become. Same thing with HIV. Many of those are used.
Yeah. We try and push
and
people
so
towards
forth.
stack when that happens. A lot of regulators just throw in E. coli, and we try to call them up, be like, just please restrict this to stack E. coli because those ones make toxins and the other one's just too ubiquitous.
Yep. So what we have managed to do is utilize a lactobacillic acid bacteria serum from Korean natural farming to treat aquaponic systems in order to eliminate those non-human pathogenic E. coli. And I think that in the future, you're going to start to see some of these probiotic practices become part of food safety practices in a way that you're seeing like regular, clean sprays used in rooms, the same, that same kind of application,
Right.
but in a probiotic way. And now that we're starting to understand the importance of some of their secondary metabolites and exudates and other things in terms of cleanliness.
I've heard of people having success on that route, which is like chlorine dioxide or whatever, like clean the grow in advance as opposed to trying to clean it after the fact. These decontamination things aren't rock solid, and it's better to strike the root and actually just have a growing environment that isn't loaded with mold growing in the walls, right?
We've treated 4 facilities for that non-human pathogenic E. coli and lettuce facilities so far. Well, 3 lettuce facilities and 1 cannabis facility, and 100% after 30 days undetectable. So
that's awesome. And so this is just a bacillus spray, effectively.
That lactobacillus, basically using kefir and air collected lactobacillus combined to treat that at a 1 to 1,000 ratio.
Interesting. Does it just outcompete E. coli, or is it actually expressing certain?
It outcompetes it and consumes it. So it feeds on the same food sources, and then it also will directly consume it. At least under a microscope, many species of lactobacillus will actively feed on it.
Oh, that's great. Oh, yeah. There you go. Now, problem with that is you might pop hot for on a tack test.
Oh, it'll absolutely grow on agar. So you're not going to pass a total CFUs, but
yeah.
Your stuff will at least be consumable, at least in a healthy
right.
Labs ain't gonna hurt you, that's what your cheese and your butter is. Just for the
Yeah, we have that going on in Cubensis. There's a temptation for people to run total yeast and mold tests on it. We keep telling them like it's always going to fail. It's a mold. So it's kind of like testing cheese for bacteria and mold.
So one of the other things I wanted to ask you about, because I know you were doing some work on it, is testing some of the microbiota of the root system. Can you touch on that? Because I know it's something that I know you're working on.
Well, what I would like to do on that is check for these viroids. Obviously, we found those in the roots, but I'm kind of curious whether they're making it into the mycelium down there, which is a harder thing to extract and get to. Yeah, soil DNA preps are hard. So it's very difficult to get microbiomes out of soils. People do it, but it's usually requires a little bit of surgery on your DNA isolation to get rid of the humic acid that's in there. We have not run a lot of soil microbiome studies. Actually, we've mostly been dealing with microbiome studies on flowers and studying, like usually whenever there's a discrepancy between our PCR kits and some other method, we end up sequencing to figure out what's going on. Sometimes it's on plates, sometimes it's whole genome approach, and we always learn something in those. Each one of those, that whole pathogenic enterobacteria study was sourced out of one of those things. And then we had another paper that published that Zamir Punja reviewed, and I think Cindy also reviewed on that went over the work in Michigan, which was interesting. We did a lot of sequencing there too, just to see every single microbe that ended up on the plate. We sequenced it and figured out what it is. But there are some things that are we now have a trend that the most recent paper kind of shows you. I think it goes through about 160 or so microbiomes, and you can that paper has links to all the microbiomes. That's probably a good place to start. You can go through there and see what are the different pathogens showing up in all these different microbiomes. But some of the, like Pantoa is one that shows up quite a bit. That's one of the enteros that we see probably most frequently in cannabis. And it's not much of a human. There are some documented cases of it getting into people's cuts. Like if you're a trimmer or something, you might want to watch out for this. But in which case it can cause some human ailments. But it's more of a plant pathogen than a human one. But that one's quite prevalent. I gotta go back through the data to see what the next ones are. But the penicillium species are all starting to resonate. That there are multiple different jurisdictions and multiple different plants. We're finding Penicillium citrinum, Penicillium paxilli are some of the most frequently found ones. So those are two. No one's really tracking those. New York was talking about tracking those, but New York's kind of it seems like they're I don't know. They've been talking about doing it for a long time, and it just hasn't happened. But those are two that would get picked up by a total yeast and mold test, but they're not targeted by any species specific testing right now. They do make mycotoxins, and one of which has been published to interact with CBD. But at what levels and do they make it on cannabis? We don't know that yet. It's just that they tend to make mycotoxins in saprophytic states, which I don't think the plant ever really sees. So no one's picked up the actual toxin per se on the plant.
That's really cool, and thanks for sharing that with us as far as those different secondary compounds. So, is there anything else you want to tell us a little bit about some of the other things that you're working on right now? I know you're always cooking up different stuff and working on different testing things. And what are some of the things that people can kind of look forward to? I know you have the conference coming up and some of the other stuff, but what are some of the other different things that people can kind of look forward to this year?
Well, there'll be a rollout of more of these virus tests to try and help people on that front. We have this feeling if people are going to start moving clones around, we'll probably see more intermixing of those things. And then we are continuing we're going to be rolling out some new. We're putting the total test right now through AOAC, so there'll be some changes there. Those are going to come out and be calibrated to the NSI standards that are out in the marketplace. In the past, they were benchmarked to a particular plating type, which is not how things are necessarily moving forward. Everyone wants standards to be locked down, one particular provider. So those are in place now. Those will be coming out. We've been doing a little bit more work that Yvonne's going to present at CannMed on live dead detection. We have a tool out there now that does it, but it's not totally compatible with all of our kits. And so we've been doing some work to make it compatible with our most recent total yeast and mold updates. So that should be interesting, because that will give us the ability to get rid of any DNA that's not inside of a living cell and not inflate the PCR values. I think that's probably the one complaint some people have about PCRs. It can be so damn sensitive. It can pick up stuff that's no longer viable after you've treated it per se. And there's some truth to that. We found that in most cases it's the media selection that you're using that's throwing the numbers off, but there are cases where you can actually zap the hell out of the cannabis flower and kill some of the microbes, and we will still pick them up and plating won't. We're working on ways to basically minimize that effect, which is an update that's coming this year. And then I'm looking forward to doing more of the genetics on the plant. There's a big pickup in utilization on Cannapedia lately, so a lot more people have been running stuff through there. We're playing around sequencing a bunch of skunk plants as well to try and see if there's any genetics there that might predict the thiol production that Ian published. I think hopefully we'll see him at CannMed. I don't know if he's going to CannMed, but he's in that part of California. So, but I don't know if you caught that paper, but they were demonstrating the skunk samples are producing some of these thiols, and we don't have any clue what part of the genome is responsible for that. So we're hoping we might be able to make some progress on sequencing some of those plants and building a better repository of information to try and sort that out.
Do you do any work with any phenols or any alcohol bonded cannabinoids or any of those types of things specifically at all? I know there's also another path of newer research recently. No, I haven't. No, tell me more about them. The last cannabinoid that hit my radar was like cannabimovan and I think THCP. So I haven't. The reason why I asked is because things like CBT, which is a alcohol bonded cannabinoid, is directly correlated with powdery mildew expression. So if you have powdery mildew, CBT will hyperexpress.
In fact, really?
I
thought. Why was I
West
thinking?
was talking about that?
I thought CBT was like is that made natively by the plant or is it get modified after the fact?
No. This is from the plant. There's CBT and CBL, I think, were the two that they were finding in high in powdery mildew positive plants.
Oh, that's fascinating. For some reason I thought those were like oxidized or something. But okay, yeah, that's cool. No, I hadn't seen much work on that. I mean, obviously we've been seeing a lot more of the variants come through the dispensaries, which is great to see. Those are starting to get products out there that have, CBGV and CBDV and THCV, but I haven't seen really anyone track those.
Yeah, the variants are interesting because they're tied to a water stress hormone, at least as best I can tell from our testing. But we managed to hit 17% in some of our internal testing.
Now, were you able to get rid of the pentyl version completely, or does it still have some THC?
No, it still has it still had regular THC and THCA and all the rest, but that
probably
was
like 5%, right?
So we had an issue with a problem with one of our facilities. Actually, it was my ex-girlfriend's house. She had a big indoor facility in her garage, and one of the sections of the aquaponic beds was on a separate loop. And we had an issue with the timer on the pump, and it ended up watering it in a weird way. And that ended up causing this hyper expression in it. Because when we did the testing on it, we're like, what the fuck is different about that? Because they're all the same clones from the same stock material in the whole room. And then we ended up doing further testing on that. So it's something that I want to flesh out a little bit more. We've done some repeated testing on it, and it certainly increases expression, but it's something I want to do a little bit more before we start. I need to find someone that we can do like a published paper with on that. But
do you have labs down there that are, obviously you got someone to measure it down there, but yeah, that would strike me as one that I'm not even familiar whether the standards would they'd have a perfect standard for it.
Well, the other thing too is CBDV, I think, is going to increase in demand quite significantly as people just start to understand it more too. So, a lot of these THCV is a appetite suppressant and all the rest. So.
Yeah, I did find some of those in when I was in Boulder last. They had them as like edibles, THCV and CBDV, which was I have to say I've never had that independent of the pentyl contamination. Like whenever you find it in flower, it's hard to discern the impact of THC versus THCV when it's a mixture of the two, right? Yep. But they actually had them isolated. So I think they were like capsules that had like small amounts, like 2.5 milligrams per capsule of like THC, CBDV and CBD, or CBDV and THCV. So there was no pentyls around. It was really interesting.
Can you fraction it the same way that you can fraction other cannabinoids, or is it different?
I'm assuming that's what they did they must have done some kind of fractional distillation to get the other cannabinoids out of there. And then they were able to make, capsules that were 2.5 and 2.5 of just the variants.
Yeah. So it's interesting. Yeah. And someone says in chat Durban. Yeah, it was a Durban cross that we actually had that 17%. So at Jamie's house. So that's absolutely it.
Oh, wild. Well, that should be exciting to keep an eye on this year. Yeah, let me know if you've got anything there so you don't think that plant's you've got clones, the plant's not shown up before until you stressed it in some way with the water.
Yeah, it had to do with the stress. Yeah, 100%. And same thing too, like the CBDV was hitting up on that too. I'd have to dig up the COA, or if I don't have it, I'm sure she still has it. So,
I mean, one thing we did find with it was Doug's variant, I think. We sequenced that one. We were poking around Cannapedia and noticed that there's about 15 or 1,600 cultivars in there, and there's about 9 of them that were labeled CBG plants, but they were labeled as type 3 plants, but they actually had a THC gene in them. Right. And normally our Cannapedia looks for the copy number of the gene, and it saw THC there. So it classified them incorrectly as type 1s. And in the name of the strain was like CBD Queen or CBG. So we're like, something's wrong here. Dug through all the data and found that there's this variant in there in the THC gene. And we even wrote up a little preprint about this, but it turned out Seth Crawford's group actually published about this during the pandemic, and we missed it. But they found the same thing, that this other variant in THC synthase known as, it's S355N, shuts down THC synthesis. Is if it's present with P3 through 3R. And you can go through Cannapedia and find these things. There's like 9 of them in there, but one of them was Doug Varen's. And the interesting thing about Doug's Varen is it had one of the copies knocked out and the other one that was working. All right. So it made THC, THCV, but since it only had one copy of the gene, it also made CBG. Like it couldn't fully process all the CBG because one of its genes was knocked out from this S355N variant that was in there. It was basically haploid for it or a heterozygous. Where that was kind of cool to see. That just the number of functional genes are going to probably change a role in the CBG expression, or how much of it gets converted.
Yeah, no, that's really interesting. I never heard about anything about that, and it makes me wonder if there's different pathogens that could, affect that too,
Yeah, probably. It's not something that, that sample we didn't have, that was like, we got that out of a dispensary in California. So we didn't have any live tissue to work with on that one. That one we just sequenced off a flower.
Yeah, no, I especially with the powdery mildew increasing CBT or CBL, it always made me wonder if we'll have some kind of either a cultured or GMO'd microbe that we spray on our plants to boost, expression of X, Y, and Z, 20 years in the future.
Yeah, I'm kind of also curious to see how all these triploids play out. They're home seeds, right?
And, I think a lot of people don't know about them or don't understand what that it is. It's something that was kind of only really announced recently in terms of the commercial market. Do you want to kind of give people a quick 101 on that?
I'll probably murder the topic because this isn't my work, but this came out of Seth's actually. I think he's presenting on this at CannMed, but you use colchicine to get genome doublings. You then flow sort those cells because it doesn't happen in all the cells, and then you have some cells that are now genome duplication. So they're basically not triploids, but they have 4 copies. If you cross that, I'm sorry, a tetraploid, the tetraploid. So if you cross those with the diploid, you get triploids, and the triploids are sterile. And so you can now make hemp that grows outside that's not going to get contaminated by your neighbor's type 1 plant. So really, really cool move. And I, they've reported the flowers and yields a little bit larger. And that's on the hemp side. I think a presentation I saw at the EMO conference from Dark Heart is they weren't yet certain on the yield, but they're definitely getting the sterility. They did this on type 1 plants, and repeated some of that work. And they've got, and they also noticed that a couple of them were naturally triploid. That was kind of interesting. I think, I'm forgetting the names of the cultivars that were naturally triploid, but I think Mac 1 was one of them. And that one's known to, you can only clone it. It's known to be sterile. So that's kind of an interesting finding out. So if they're in fact bigger and they don't get pollinated, that could be very useful for a variety of outdoor grows. But I'm kind of curious what that does to all the terp expression, everything else, because the genome doubling isn't just going to obviously the sex chromosomes get affected, but so do all the other genes. So do they produce more THC? Do they produce different terpene profiles? Like a lot of those things are, I'm sure, are still just getting worked out from their perspective. And then the other question is, what about pathogen response? Are the germination rates the same? I mean, that's something that, they've got some numbers on that, but I haven't. It's too early, I think, to tell how well they're going to replicate everywhere else in the world. But it does look like a nice, exciting new tool we've got in the tool belt.
It's super exciting, and yeah, I know I never thought about that and all the different secondary implications that it might have. Well, is there anything else you wanted to mention here? I wanted to throw up your Instagram and some of your other links here for people that want to follow you. You can check it out, Kevin_McKernan, MCKERNAN_73 for listening to you in the audio version on Instagram. And then you can find him over at medicinalgenomics.com. and CANNMED events.com. And then his podcast is canmedevents.com/coffee-talk.
Excellent. No, thanks for having me on. Really appreciate it. Looking forward to. Feel free to reach out if folks have more questions.
Oh yeah, always love having you on. Always love learning about the new different things that you're working on. Really excited to learn more about all the different mushroom genomic information that you're putting out there, and always following your Instagram for some of the best new info on plant genetics and many other things. And I really appreciate what you're doing, and lots of good info. Definitely want to get into your podcast here in the future. And yeah, thanks a lot for all that you're doing. And if people are looking for different test kits and stuff, definitely check out Medicinal Genomics. They're definitely one of the best sources of plant pathogen testing and all kinds of other great stuff.
Excellent. Thank you so much. Looking forward to this. I learned so much in every one of these, so we got to do it again.
Thanks a lot, man. Always love having you.
Yeah. Cheers.
Cheers. Take care.
Cheers, man.
Well, that was wonderful. Always love having Kevin on. How you doing, Charlie?
Good. How you doing, Steve? How you doing, Portland?
I'm
Good.
doing great.
That was incredibly informative. I mean, that was just a plethora of freaking information to digest. It was fantastic. All the things that they can do now.
He's definitely.
All right, you're making me blush, so I'm going to run away now.
Oh, sorry. Hey, you're not supposed to be here. Wait a second.
I know. I'm sorry. What are you doing? You're trimming plants on me?
I'm stroking that ego, bro. Just gotta stroke that ego, man.
All right, I'm out of here.
Cheers,
You guys,
bro.
thanks.
Much appreciated.
Yep.
I know.
Thanks.
Kevin is truly top of the field when it comes to cannabis genomics or mushroom genomics. Few can rival the experience that he has. There's few people that know more about either of those topics, and I mean, I think that was the fourth time he's been on the show, or fifth time he's been on the show, and I think you guys can see why. He's just a wonderful guest, and really has done a lot of the hard work that the rest of the industry is going to really rely on when it comes to these genetic tests and alleles and figuring out a lot of these mysteries that we've been running into when it comes to different pathogen expressions. Just like you're saying too, I'm a firm believer in that this viral stuff is going to become a bigger issue as we go forward, and people start to understand more about what's going on. So
Yeah, and they're gonna constantly mutate and change, and to stay on top of it like that, they're gonna find the defense to everything that we throw at them, and they're gonna constantly start evolving. Just like they've always been doing, but even more prevalent as the industry gets going, because there's going to be more and more of it. It's going to come to the forefront. I mean, it's a shame that we haven't been able to do this for decades. It's just, it's only been in the last couple of decades that we've been able to really start figuring out all this stuff. I mean, I know it's been in the mainstream commercial aggie, some of the stuff, but some of it is definitely cannabis specific, especially when it comes to yields and everything else, I mean, a little bit different than just the commercial aggie and growing a bunch of corn or a bunch of wheat,
Yeah, and having those types of just like he's saying, having those types of community-based data inputs is what's going to become critical for finding this stuff out. I know anytime I find anything weird, Kevin's always happy to send me a genetic kit and to get him some weird samples. I'll send him some pictures, and if it's something that he hasn't seen before, he's usually pretty cool about it. So certainly it's definitely worth it if you're a commercial producer to get your stuff sampled, and if you're trying to do any type of protections long term as well, you can do genetic sampling through them. Although most of the stuff that they do is entered in through Cannapedia for open source stuff. But hey, if you do have something, it is a resource that you can utilize.
Yeah, absolutely.
So
what's his background? Where did he go to school? I'm just yes, sorry.
Go ahead. Sorry.
Yeah, I'm just trimming some stuff that I took down about a week or so ago, maybe a little bit more. It dried a little bit quick, so we have a lot of dry air around here lately, even though we've had some rainy days. But it really hasn't. The humidity levels in the air have not been all that great. So I slowed it down a little bit, and now I just want to jar it up with a little bit of fresh stuff just to make sure that it stays slowed down. I don't want it to. It's actually, this stuff's probably been about 10 days now. It's not quite. I have, some of the smaller ones are snapping, the bigger ones aren't. So I want to get it more evenly spread out, the moisture. So I'm just kind of just doing some maintenance on my harvest.
What would
More than
somebody
anything
do
else.
if they had their
So,
bud in that type of state? What's your protocol when you do have about a 50-50 like that?
get it all jarred up as soon as you can, and then babysit it for the first 24 to 48 hours. I'll check it every maybe 6 to 8 to 12 hours, depending upon how moist the real big ones are compared to the smaller ones. But I'll dump it out again, check everything out, let it sit out for about a half an hour, see where it stands, and then jar it up again. Just keep constantly going back and forth. I really don't use the Bevitas or anything like that because I have too much. It's just crazy to have to buy all those, so I don't do that. I don't let anything get to the point where it can't get just by putting a fresh bud in or two, to have it come back a little bit if it gets a little bit too dry. I do have some people though that no matter how I cure it, they'll still let it sit out for, they'll let a couple ounces sit out for at least 24 to 48 hours. They want it super dry, and I'm kind of the opposite. I don't want it super dry. I mean, obviously I want it to break up just perfect, but I don't want it to be, I don't want it to just, turn into powder. I have some patients that just love it to turn into powder. So, and then they only smoke it. It's not as though that they process it in any other way, like edibles or, whatever. They just, but that's just the way they like it. But so, yeah, I'll just babysit it for I'll have to just babysit it for a while just to make sure that I'm getting that even across the board with all my buds. And it just takes time. It takes patience and time. That's really what it takes, which can be a real pain in the ass sometimes. But if you want to get if you want to, there's nothing worse than spending all that time on a crop and then all this, you have it get too dried out, and the terpenes and even the effect, it's nothing like it should be. Some of these plants I've had for 10 years, so I know what they should be doing for me. So, yeah, I mean, it's not like there's anything special or scientific about it. You just gotta be, you just gotta stay on it, So, and I've been so busy lately, I just haven't had a chance to. My wife usually does, that's what she does at night. She takes care of the trimming and the curing. She's gotten very good over it after the last 12 years or so. We've been together about 15 years, but she's been doing it since I got out of the hospital. I've been trying to, teach her my way anyway. And she's gotten very, very acute at it, I gotta say. So, because she does most of the trimming and the curing. And she's been busy. I've been busy. So, something's got to give. And my daughter's been home all week for vacation, school vacation. So I kind of neglected a couple of trays that I had going, just drying. So, but yeah, I'm actually going to try smoking some of this in a few minutes. This is just this is the stuff that I had at the place that I show, the main place. I got another 4K upstairs. Lights just came on, and I've been having a problem there. I had I haven't run that room in over a year and a half, so I checked for light leaks because I'm running the lights at night, so I wanted to make sure everything was still all set. Ended up having a slight little pinhole leak in one of my ducts coming in, and my intake, and that lasted about 2 days. And I finally caught it, but then one of my timers didn't go off when it was supposed to go off, and I think somebody may have nudged it, knocked it, whatever, brushed up against it, and I didn't realize it until yesterday.
What's better is switching over to those Amazon smart plugs, because those don't, and this is just Marty's got some that have been running for 3 or 4 years. I've never had a off the shelf timer last 3 or 4 years in all the years I've done stuff. Right?
Yeah,
So they're already outlasting the stuff. So some of those
I
smart
believe it.
digital ones are worth the money.
Yeah. You know what I used to use?
you can use your cell phone and monitor, especially I know you work in
really?
multiple grows. You can have
Yeah,
each
yeah.
room set up on its own thing. You can monitor temperature and what's on, what's off. You
Oh,
basically,
yeah.
what you do is you plug every single electrical device into it. You assign it a name and then a
Right.
power draw, and then you can say, hey, well, why is the fan working so hard? The filter needs to get cleaned. Or hey,
Right,
why is
right.
the water pump working extra hard? Oh, the pre-filter needs to get cleaned. Or it just makes it, especially when I work on large aquaponics facilities and I have multiple rooms and I have 2 or 3 or 4 pumps running and I have backup pumps. So what happens is if they have an emergency, I can see that the main pump is down. I can shut off that pump, turn the solenoid on, switch, close the one solenoid, open the other one, and turn the backup pump on without any employee having to do anything remotely.
Oh, that's
So
beautiful.
it means make sure that there's like an insurance policy where the owner knows that I can make sure that even in a disaster, I can, remotely, regardless of where I am, take control of the situation. You know what I mean? So,
Yeah. My God, yeah. Why the hell haven't I even
but
heard
you
of
can
these?
do,
For
even on
Christ's
the
sakes.
basic level, for like $200, you can rig up all your, between $200 and $400, depending on how many electrical plugs you need. You can rig up a room and have it be completely. And Marty did a whole
Yeah,
guide
yeah.
on YouTube channel over APMeds on how to rig that up on Amazon with your Amazon smart
Mhm.
plugs.
Well, yeah, I'm gonna be all over that tomorrow. No question about it. Yeah, I mean, the best plugs I've ever used were, funny enough, were the GE dual digitals, and I still have some running to this day, 10 years later. Those ones from Hydro Farm or Sunlight Supply, I bought, I don't know, maybe 10 once about 5 years ago. I think out of the 10, I think I still might have 4. That's it. And the other ones shit the bed within, a couple of months. Something that I always had to stay on top of because I knew they were going to kind of shit the bed. So those GE plugs that I use, they're fantastic.
What
And
you
they're just
do is
simple. Home Depot,
you
$6.
get the weather station units from Amazon, and you put one in the grow, and then you put your temperature sensors wherever you want to put them. They're separate. You got all these. You got to keep the batteries in them, right? So you got to get some
Sure,
rechargeable batteries or whatever, like whatever. But
sure.
you set all your sensors up, and then your air temperature sensors. You set all your plugs up, and then all of those are controlling it. You can set it all up so that like fans turn on if the temperature gets too high. It automatically turns on extra fans, or you can rig it up however that
Sure.
you want with different high and low temperature settings and whatever other metrics that you want to set it to do.
Yeah, I'm gonna look at it because I'm actually the room upstairs isn't even kind of finished. I don't have it completely dialed in. It's I'm keeping my temp. I'm just getting my temperature in gear, my parameters, and they're starting to stay. The time is that they were in 10 days or so. All of a sudden, my wife cleans out a hallway. One of the times is because I can't run all my cords because I'm at home here, so I can't run all my cords the way obviously the way I would love to. So I've got to drag friggin' extension cords everywhere. I get the 14 or 12 gauge extension cords to make sure I have plenty of power going through those friggin' wires. And then yeah, she cleaned out the hallway and she must have brushed the on off button and it just stayed on. So I didn't catch it for 2 days. Now I got my Hell's Angel, one of my Hell's Angels in there. It's looking funky, man. Like it doesn't know which way it's going. And I'm like, I know I'm gonna have to take this one down. I'm just hoping the rest of them is 23. I don't have to take the rest of them down.
I have
That's
a,
for sure.
I have a question. What's your opinion then? Because it seems like this might be a similar scenario. I was listening to Ross to Jeff this week on I don't remember which show it was. I think it was a Dude Grows show, but it might have been on one of the other ones about him doing longer light times at the last week or two of flower in order to boost flower production. What are your
Yeah.
thoughts on that?
Yeah. I don't know. I mean, once you're past a certain point, it's worth trying. I've never done it. I've always run, well, at least for the last 8-10 years, I've always run, 13 and 11, 13 off, 11 on in my cycle. and never had a problem. I find that some strains initiate flower a little bit, a few days, 4 or 5 days earlier, some of them, depending upon which strain it is. But I've never run it longer. Maybe you're going to get more production, but I also think that you're going to, and I'm just saying this obviously shooting from the hip, but I would think that maybe, some of those strains, man, and they might not like it. And how quickly they're going to, I don't know how long you're going to do it for either. You might, if you do it for a day or two, or even a week, maybe, I don't know if you're going to have any problems. Anything longer than that, I think that you could actually end up with fluffier bud, because that plant is going to want to kick right back into veg if it's really sensitive, if it's sensitive genetics, like any cookie strain or anything like that. I don't know if I try it with any, I've always found my cookie strains to be highly sensitive to any kind of light cycle or changes. And, yeah, yeah, that's one that, I don't know if I try keeping the lights on longer. I would certainly back off. Excuse me. I've backed off my lights a little bit towards the end sometimes to get the cooler temps, maybe to bring out some color, or to bring those terps right out. So my temps not, and the terps on is, because I can run my lights pretty close to my plants. I'll really push them. But so in the end, I can back off. I don't necessarily, I haven't for years anyway, shut my lights off for days before I start harvest. I know some people do that, and that's fine. I've never had any problems. I mean, I've never heard of anybody having any problems with that. But to put my lights on, I mean, the worst you might get is, I mean, it's too far into the cycle to have anything to really freaking produce a seed. If it does produce a seed, or a herm, a sac, it's so late in the cycle that most likely you're going to get it down before it could really do any damage to your harvest.
Yeah,
That's kind of the way I look at it. But
it's something I would love to do a side by side on when I'm able to. It definitely got me thinking.
Yeah, I'd be really curious too. I mean, shit, if you can pump out an extra ounce of plant or something like that, just by leaving your lights on, that could be quite a significant difference when you've got hundreds of plants in a room.
Oh yeah,
that's a huge difference. Yeah, God, I would love that. Yeah, let me know when you do it. I might even try something like that if I ever have the opportunity.
Well, I'm not sure. Well, it'd be interesting to hey, if you have a tent, throw a tent up and throw that in the corner and just give it, a mirrored light schedule with one light on it or whatever. You know what I mean? And see, but give it that longer light schedule at the end. Just those last 2 weeks, throw it in there and give it that longer light schedule.
Yeah, I do actually have a 5x5 or a 4x4 hanging around. Maybe I'll throw it down in the basement and see what the hell I can do with it.
That's what I'm saying. If you're already condition, if you got anything in flower and it's getting close, just grab it the last week, 2 weeks, and toss it in the room and grab one of the lights, throw it in there, and you see what I'm saying?
Yeah, absolutely, no question.
And just to do the side by side, because Ross to Jeff was swearing by it, and I think it makes sense. Especially that late too, just like you're saying. By the time it had a chance to light herm, you're going to be chopping the thing down. It's not even going to have dumped pollen yet, better yet, fertilized anything. So,
yeah. Dude grows tonight. Actually, one of the headlines was pollination late in flower. That was kind of the headline I saw of his show. I didn't get into the show far enough because you came on, so I kind of switched over. But I was curious to see what they had to say about that. If you did get pollination late in flower, I don't think it would have any. As long as it's so close to harvest, wouldn't do anything.
Have you done any seed production? What's your preferred method?
No, I've never done any seed production. I've done quite a bit of testing over the years for other people, but I never have. Lately, though, I will say with what Fumi's been doing and other people, and I've been very curious to maybe start, especially in Tommy Trikes, testing out some of these strains that I've had around that are truly favorites of mine and patients, and see what I can create by crossing maybe a couple of them and see what comes up. Just be a chucker and just get my feet wet. trying a little bit of it because yeah, I would love to get into some breeding, to be honest. That would be a really fascinating area. I've just never tried ever. I've always had bag seeds in the beginning, you know. I mean, that's what I started with bag seed. I mean, we obviously have, when we gorilla grew, we'd always have that friggin' male plant here and there that would be too close to our females, and you know, half a plant would get, depending on how the wind was, sometimes we get half a plant that was seeded and half a plant that wasn't. But yeah, I'd love to. I would love to, especially with this blueberry fuel. This blueberry fuel I've had around forever, and it still is one of my number one favorites. It's like I gotta have this available every week, no matter what, even if I don't have anything new. You know, they want to try something new, but then they always go back to this after a couple of weeks. Though I just took down that the other one too from Exotics I got was that lemon freeze pop. Heavy indica. I smoked it last night for the first time on the show. Oh my God. An hour after the show, I was sitting down, fell asleep sitting up again. Because it hit me so hard. Because I'm not a huge indica guy. But yeah, I would like to see maybe I could do something like that. I mean, and it's got beautiful lime color to it. The smell is fantastic. And then you know, see what I can do with something like that. I got my seed collection downstairs, and I got to go through it, pop some seeds, see what I can get. You know, I had a Durban poison 2 years ago. I had 3 seeds that somebody gave me from New York, and it was a true Durban to, you know, structure, growth, everything. I mean, things stretched from friggin' to the sky, and you know, made those huge colas, but very airy. You know, straight equatorial. There's no question about it. But the terps, I can see why it's run mostly to process and put into cartridges and oils and stuff like that, because the terpene profile is just absolutely fantastic. But you know, if I can crack some seeds and find something like that, because I got 1 male out of 3 seeds. I got 1 female or something like that. I can't remember. I know I got a male that looked beautiful in veg. Never flowered it out, and I should have taken the opportunity at the time, but I just didn't have the time. So I just kind of chopped him down. After I kept him around for a few months first, because I was like hoping to maybe do something with them. But if I could find something like that, and then, you know, start doing some experiments with crosses just in a tent, like you say, that's always been my if I was going to do it, that's the way I do it. And I just put it downstairs in my basement, away from everything.
The nice thing about the Durbans is that they're super heat tolerant. I mean, they're one of the ones I always often recommend to people outdoors in Oklahoma, because they don't care if it gets 105, 110.
Yeah.
As long as you give them a good amount of water in the evenings, they're perfectly fine.
Yeah, I could crank the one I had. I could crank it literally right up on top of my light, and it didn't burn, and it didn't foxtail. I mean, it just loved it almost, it seemed. And I had it practically the top of the plant touching the glass on my light, and it didn't even blink an eye. And middle of the summer, I think my AC went out one day while they were doing electrical work in the building, and I forget how it happened, and my AC went off, and it was like 104 in there. It was the only plant that kind of laughed at it. Thankfully, my other plants were like, you know, help me, please cool me down quick. But you know, nothing drastic happened, because it was only for about a day. But yeah, those that's the one thing I did find. That was the only time I had a real true Durban poison, and I saw what it could do. Fantastic plant, man. I will say that. Just the flower wasn't such a huge hit. I should have just processed it all. I had my guy process it all. I got a good guy that processes everything that I get. Remedies. Remy, this kid Remy from around here. He's won a couple of he actually came in second at one of the Emerald Cups, I believe it was, out in freaking California. This was in 2015 or something. But he makes this beautiful stuff now, and it's all solventless, if I'm not mistaken. And I should have had him run it. I got so much for him to run. I won't let anybody else touch my stuff, so I got stuff stashed in my freaking mini fridge, waiting for him to get up and running again. I guess he's doing it for some one of the big processors now, but he's looking for a space for his own. He's got, last I knew anyway, he had like 6 tubes. He actually got raided a few years ago. He was totally legal too. I mean, he got off the case, and it cost him about 10 grand. for the lawyer and everything else. But a lot of people helped him out. I helped him out as much as I could too, just from finances to making sure he had medicine and didn't run out. But he had 6 1-pound tubes running. 6 1-pounds or half pounds, I can't remember. But either way, they were good size friggin' tubes that he was running. And I believe he was doing the CO2 dry ice. That's what it was. He was doing the dry ice extraction, if I'm not mistaken. But yeah, he was like a friggin' machine. So I wish I had him back then, and I could have freaking had him in my Durban. That would have been a beautiful thing. But yeah, I'm just trying to get this place upstairs. I'm really tweaked, man. I hope the rest of my plants fared with that light leak.
What cultivars are you growing these days?
I have that lemon freeze pop from Exotics. I have apple water, which I just took down the first run of. My wife is trimming that right now. And then I have the Hell's Angel that I got from that Hell's Angel OG, the biker cut that I got from Swerve years and years ago, 8 years ago anyway. I still have that around because that's still well liked. Plus, I like it. It's one of my favorites. And then I have this Blueberry Fuel that I sifted myself. The other ones are from clones. I just mentioned the apple water and the lemon freeze pop. And then I have the Huckleberry cookies. I sifted that from seed. That one's going to get the cut, I think, though, because it's going to get the X because the X rather, because it's just too much leaf, man. It really produces a lot of friggin' vegetative matter around those buds. So I think that I'm going to get rid of that one because it takes forever to trim it. And then I also have I have the GMO. I have I got the real cut of the GMO. I have what else do I have? I have the garlic breath from Thug Pug, I believe that is, and then I have Tropicana cookies, the beautiful cut that I got from the same the only guy I get cuts from. I don't get cuts from just anybody. I just get them from one guy, and he sifts a few hundred seeds at a time, and he'll pick out the keepers. And so I have Tropicana cookies. Smells and tastes just like a freaking sun-kissed orange. And then I have, and it's really, really dark. Like you'd almost think it was black. And then what else do I have? I'm missing something in the mix here that I have. I can't remember off the top of my head. There's something though I'm missing. And then I have, and then of course the Blueberry Fuel, if I didn't mention that. Sour apple I have. That's another one, sour apple. And then, I had a bunch of ocean grown stuff, a few years back, like Bewitched. I actually had Love Potion number 99 that I got from PH Nerd. And that was fantastic. And then, but I had a lot of stuff. And then they started making some strains, and I was testing them, and everything I was getting was bunk. I even got some of Veda's Jawa Pie. I cracked 10 seeds. I got 2 that I thought might be keepers, but I ran them like 3-4 times, and I was like, oh, this thing isn't anything special. I got to take this thing down. I find that a lot. With seeds. You really have to sift quite a few sometimes, depending upon the genetics, obviously, but you really got to sift because I can't. It's the way the market's going now. You got to have not just a cut above, man. You got to have a cut above, because everything I was just reading today too about how the market's taking a dip and how investors are losing money left and right because they were hoping that things were going to change with the Congress and everything. Since everything was democratically run, they thought it was going to be a friggin' big shift, and finally the federal banking system was going to be open. And of course, it's gone nowhere. So they're trying to say that the black market's still thriving, which it isn't. It's thriving, but the prices aren't there. But I don't know who they interview for people, but they're saying that some of the comments, actually, I think it was that I was reading on the article, they were saying how they can pay cheaper prices on the street than they can in the stores, and they're getting most of the time they're getting better stuff than they are in the stores. Now I know Massachusetts is really up their game over the last year and a half. Things have gotten a lot better, especially when it comes to choice of a flower. Flower was going so fast that you'd walk into a store and you'd maybe have 4 strains to pick from, and out of those 4 strains, one might be, halfway decent, but they wanted an arm and leg for it. They wanted the full, $450, $500 for an ounce. That's before taxes. So you're paying close to $550, $600 for a freaking ounce of weed. That's absurd. That's just absurd. How can anybody afford that? And even on the medical side, how can you afford that, man? Just insane. I give so much away. Just so much away. Anybody needs anything, just contact me, man. Yeah, take it. You need it, man. You need it. Just call me. Just, email me. Do whatever. DM me on Instagram. I do check my DMs.
I got a friend of mine makes a bunch of rosin, and he saves all the pucks and cooks it down into butter and into olive oil or coconut oil. And,
Nice.
Keeps it, and then whenever there's a little old lady or just someone's like, "Oh, my dad's sick," it's like, here's 6 bottles. Let me know when you're empty.
Yeah. Yeah. My godfather right now has Parkinson's, and I know it would help him. And he's just so old school between he and my aunt, and even his kids. I don't think that I can't even bring it up to him. I can't even bring the subject up, which is a shame, because I really think that it would. And he's really progressively gotten worse. I mean, I haven't even seen him over a year and a half because he just doesn't want to be seen. I email him and I email, his kids. We're really close. But just to even bring up the subject is really, it's like I'd have to tiptoe around it. And I don't even know how receptive they would be about it, because they are so conservative. Same with my father. My father has this disease in his mouth. We finally found the name out, actually. Easter, it's called Magin, MEGEN or MAGEN, and it can start in your mid years. It started with my father in his late 30s, early 40s, but it can be brought about by stress, physical, emotional stress, and it's where his whole mouth, from the bottom of his nose, of his septum, and his tongue, he completely loses control. It's tough for him to talk. It's tough for him to eat, and it actually interfered with some of when he was at Hancock. He ran the real estate department for years, for almost 40 years. But it, he could have become the COO of the department, and they wouldn't give it to him because of his disease in his mouth. And they've thrown everything at him, everything from from Valium, diazepam to Clon, and he hasn't taken them. I don't think he ever took. They gave him 500 Valium one time. Now this was back in the early 80s. And he didn't take him. He still has them in his friggin' drawer, and he and I joke around about it. He's like, "Hey, Bill, can you make some money?" and he wouldn't, and he doesn't take it. But I wish I could approach him on this. And to be honest, if it wasn't for my stepmother, I could approach him on it and ask him if he'd be willing to try something that might help relax those muscles, that would help him, even eat. Just a shame, man, how people have really stuck on stigma, And it's not my father, it's my stepmother, She is just so, oh my God. Easter was just, it's like you have to, God love her. She came into a family with 3 young kids when she adopted us and married my father and all that stuff. But, she was just so ruthless. She brought the convent with her, excuse me. And, you just can't bring up anything. Well, I shouldn't say that. She has a thing. She has a thing for me. She doesn't like me that much. And because I'm a complete 180 of what she is. And but I wish I could. The funny thing is I could talk to my father about it privately. But every time I get any kind of privacy with my father, she's got one eye on me, because she doesn't want me talking and getting private conversations, even with my brothers. She doesn't like it. So she came in and started. She started on Sunday. I was talking to my youngest brother, who's her only kid, through the marriage. And as soon as she saw it, he and I were alone talking. She came beelining it right into the kitchen just to see what we were talking about. So, and there's my brother all stressed out, man, crying on my shoulder about shit, And it's like, just can you just leave us alone for a few minutes? Do you mind? But I just wish, bottom line is I wish I could help my father, at least talk to him, because he's not anti he's not anti-pot. I mean, he admitted to me back in the 70s how when he first started with Hancock, he was on some business trip. I can't remember if it was California or Florida, because Hancock owned all the orange groves, and he was in a convertible with a bunch of guys, and one of them was a younger guy and probably my father's age at the time. And he pulled out a big bag of weed. My father took a look at it, smelled it, and all that stuff. So, my father's not, Mr. Tight Ass like that, but just my stepmother is. But, he's 84. He's got bursitis really bad. I know some of these would help with that inflammation. He's got bursitis in his hip really, really bad. Like he can't, sometimes he falls, he begins to fall over when he takes a few steps, He's going to be 85 years old this year. So, I'd like to see his quality of life get better before it ends. He's my only real parent left. So
Yeah, I know when I visited my pops.
That's why when you say education, that's why whenever you say we need to educate the public, I just my ears perk up. Like yes, like these people need to be almost shoveled down their throat. Like get over it, will you? More people die from friggin' alcohol, drugs, and no one's ever died.
Last time I visited my dad, I left him a vape pen, and then the time before last, I left him a vape pen, and this time I left him some gummies. I figured, I'll leave them in the fridge, and I'll little note, hey, if you can't sleep one night or you're in a lot of pain, just take this. Nothing bad will happen. You'll be okay. And you'll take this, lay in bed, you'll have a good night. It'll be fine.
Have you noticed them? Have you noticed them like slowly diminishing in the fridge? Have you been back?
I don't know. No, because my dad lives in Philly, right? So I'm only there for a couple of days a year. So
yeah.
But yeah, we'll see if they're there when I go back.
It's funny. Yeah. I mean, I don't live I live about an hour and 15 minutes away from my father. I mean, he lives, blocks away from my two younger brothers down in Marshfield. But I mean, I could see him more, but I have to go through my stepmother. And it's just, oh my God, if you knew the friggin' hoops you have to jump through to just friggin' talk to my father, my God. But hey, they've been together 50 years now, so what am I gonna say?
What tips or tricks have you learned over the years? You got quite a bit of experience on the cultivation side. Tell us a couple of things you think maybe newer growers might need to know, or maybe some tricks that you can tell us to save some time or labor or other things like that.
Yeah, I don't know, pretty simple. I use the KISS method I always have my whole life, no matter what it is. Watch your plants, inspect them every day, go from top to bottom. Especially when you have a small grow, it only takes a few minutes to go through everything. Make sure you keep that airflow going. They don't have to be friggin' waving in the wind, but you want a little bit, you want them moving a little bit. So make sure you have plenty of airflow. Clean up your bottoms. Inspect when you're cleaning up your plants. Make sure you don't have any pests. The best way to do it. Take the time. It's all patience. Take the time to make sure you inspect your plants at least once a week, just to make sure you have no, anything from powdery mildew. Powdery mildew is not going to happen just in flower. It's going to happen in veg. So inspect your leaves. Take the time to inspect for bugs. You can't just look at one leaf and think, oh, I don't have any problems. You need to go to every part of the plant, top, middle, bottom, and take at least a couple of leaves from each and take the time and scope them. You're not going to see the damage until it's too late most of the time, especially if you get spider mites or even thrips. Thrips can, they're slow migrators, just like spider mites can be, but you want to catch them early, instead of make them a nuisance, not a problem. Don't spend any money on sprays. I mean, if you do, Trifecta, it can be a little pricey, but it's one of the best that I've ever used off the shelf. But make your own. Use the coriander, the garlic, the peppermints, the clove. I mean, you can make your own. Boil it up, sift it out, and then take that oil and then add, do your ratios and just use that. It's super simple and it's extremely quick, and it won't smell as bad as some of those crappy sprays you're going to buy in the store for an arm and a leg. And that's the one thing I like about Trifecta. It actually smells pretty good. So and just get to know, when it comes to soils, I've never done a living soil. Super soils, yeah. Make sure you get your dry back. Don't overwater. You're better off underwatering than you ever are overwatering. Once you overwater, you can potentially have a problem right off the bat from one overwatering. Some of those the roots start dying out, they drowned, and you're gonna have some problems. And sometimes the plant, depending upon the genetics, it'll never come back correctly. So, or in full health sometimes, because it's beating its way through those friggin' rotten roots down there. So, that's why I can't wait to try a living soil. That's what I'm gonna switch this place over. Every time I have to go to the store and buy another bottle, I run Canna. I run a very, very simple recipe. I use the Cannabis. I never use it at full strength. If you do use bottle roots, ever use them at full strength, use them at a quarter to a third strength. And then if the plant says it wants more, then you can up it, but always never give it anything that the package says. And just make sure that you're, and like I said, the big thing, one of the biggest mistakes I ever made back in the 80s when I went into it was that I was overwatering so much because, this is the biggest problem I made at first too. The biggest thing I did at first was I was giving them too much attention, overthinking things. That's probably why I was overwatering a lot back then. Let them droop a little and talk to you when they want to drink. Wait till you see them and lift your pots to make sure that you can get a gauge to when they are dry. I mean, I have my garlic breath. It doesn't like to get completely dried out. The GMO doesn't like to get completely dried out either. Though it can handle it really well if I do, because I want to get my watering schedule all aligned as much as I can for everything. So, get to know the weight of the pots too. No matter what size your pot it is, it doesn't matter. Get used to it, and then, but yeah, don't give them too much attention. Give them the right. When you do give them attention, give them the right attention. Don't overdo it, and get your watering down. That's kind of key. That's another reason why I want to go to friggin' living soil, because then I can just put the pipe in there and do a little wick feeding, just constantly have something that's going to have a flow valve that can top itself off. And, I'm a pH guy. I'll probably still pH everything, if I do, teas and everything else, but I don't even know if I'll do all that stuff. I just want to get down. I just want to get the feel for that living soil, man. I'm really kind of excited. I just don't want to have I can't afford to have a bad harvest. I gotta pay the electric bill. So I wish I could do outdoor, but I can't do outdoor. I got neighbors that just right on top of each other. So, and I don't have the time to go out and find a place to guerrilla grow. So I'm just gonna stay indoor for now. But yeah, just don't give your plants the wrong attention. When you do, give it the right attention. And watering key. Learn what they want and what they don't want. And every strain can be different. But they don't like to be wet. That's the one thing cannabis plants, they don't like to be saturated all the time. I mean, living soil, I'm not talking living soil, I'm talking cocoa super soils. They don't like to be completely saturated all the time. So just get your watering down. Pretty simple. And stay on top of the bugs, man. Stay on top of the bugs. Make them a nuisance and not a serious problem. Because we've all been there having serious problems. it's usually because I don't give them enough attention. I'm really not taking the time to really inspect my leaves and, even the stems too. Some of the stem crawlers, man, they'll crawl right off that leaf and right over the stem just to hide from you. That's about it. Nothing serious, I guess. That's about it.
You any plans for any new strains that you're going to be popping, or are you pretty settled on what you got, or do you have any kind of?
No, I'm constantly changing. Yeah, no, I constantly. I have some of the ones that I named off already have been staples in my gardens, but I usually change things up every. I'll crack seeds every 6 months, 8 months. I'll crack 4 or 5 new strains. I just haven't done it because I wasn't sure where I was going to land with that problem that I was having at that garden, and so I really haven't. I haven't changed anything up. The places that run commercially, we just get everything from clone. So, and it's supplied by the same guy I get the clones for my personal gardens. So, but yeah, I have a bag full of seeds downstairs. I'm gonna start going through and see what's up. I have actually some of Tommy's seeds that that Smash gave me when we went to that thing in New York back in whatever it was, October, whatever. And then I'm definitely going to crack some of his seeds and see what I come up with. I think it's the G13 skunk, I believe. I believe it has the skunk in it for sure, the one he gave me, because I was really excited to see the skunk in it. And I've got some archive seeds that I'm going to go through and see which ones I want to pop. I actually have a number 22 do-si-do I was thinking about, because I heard it got better as it went down, as it got older, and a little more work was put into it. I heard that the archives 22 is pretty good. I heard the original do-si-do looked really good on the vine and looked really good in the bag, but it really wasn't anything to write home about when it came to effect and taste. But I'm hoping this one might be, because I've been thinking about that one for a while. So that one's definitely one I'm going to crack. And I have so many. I mean, I got a bunch of. I have a bunch of ocean-grown ones that I might go through and see if there's anything that's worth doing. But I was with them for 10 years, so I don't know if I'm going to, or 8 years. I don't think I'm going to do anything from them, but I'm going to go through in the next couple of weeks and see what I can sift out that catches my eye. But definitely that do-si-do number 22 for sure. And then there's another one I can't think at the top of my head that I wanted to do. I do have some autos that were given to me that were supposedly they were excuse me, they were given to me but up in that convention up in New York. It's oh god, the name's going to escape me who they were made by. Very prominent name. Give me a second, mental block, but tip of my tongue. Oh god. Oh, and I didn't put a label in it either. Who made them? Watch, as soon as we're off the show, it'll come to me who made them. In fact, I think I have them right here. No, I left them at the garden last night because I left that bag there. But anyway, they're going to be the first autos. Now I do have some autos that I've had around for years, and I mean like 6, 7, 8 years. And I don't think I'm going to crack those, only because I know back then they really weren't they really hadn't been worked to where they are today. So I'm hoping the ones that were given to me. See, I'm still thinking of that name. See, it's just right there. I think the last name begins with an L. Famous breeder too, and I can't remember who the hell it is. So I think I'm gonna crack some autos and see what the hell I get out of those. And because those can go under any light cycle, if I'm not mistaken. Is that right? I can put them on 24 hours of light, or I can do a
Again, we never recommend any plant be under 24 hours. 18.6
Right.
is really the maximum we ever recommend. But there was also a question in chat. Someone said, where is it? Is regalia okay to use with living soil? Yes, regalia is perfectly fine. It's just an extract from giant knotweed, which is a pest it's actually a huge problem in the South. So they basically harvest this invasive weed, extract the juice from it, and then make a product out of it. So it's nothing inorganic about it at all. I don't think it's OMRI certified, but there's nothing inorganic about it. Maybe not quote unquote organic, but that's not my specialty.
Yeah, I almost got some of that. I almost got some of that about a year ago.
So I've used it. It didn't work, and then I moved on. So if it works for you and your particular strain of powdery mildew,
Yeah,
great.
right. Yeah.
I have not had good results, but doesn't mean that you won't. Just like we were talking about earlier with Kevin, there's so many different species of powdery mildew. It's going to this is the reason why you need to have multiple things in your toolbox. So that if something doesn't work, you're like, cool, that didn't work, we're going to move on to the next one. That didn't work, we move on to the next one until we find something that is in your toolbox that's going to work for your type. And sometimes you'll hit it on the first time, sometimes it's the second or third, sometimes it's the fifth. It just depends on what it is that you're trying to get rid of.
Yeah, good advice. Have more than one friggin' bullet in your chamber. Make sure that you can hit everything, no matter what.
Oh yeah, especially if you're trying to stay organic, going with biocontrols, having the Beret Bassiana and Asteria fumiceraceae, or what's it called now? Cordyceps fumiceraceae, I think is the new designation on it. They just changed it a couple of months ago because hey, let's be confusing. But PFR 97 is the same product at the end of the day. But alternating with those different types of probiotics will give you much better results. All right. Well, do you want to tell everybody how to find you, or if not, you know, you can find him on the Fumi show at Fumidor and the Flavors and my show. And then if you want to plug anything else, great. If not, that's fine too.
No, that's where to find me. I mean, I have videos on YouTube and I am on Instagram, but I haven't been on there for forever, you know, and I always say I'm going to get back on, but I don't have time. Last time I was on Instagram, I was spending sometimes 4 hours, 4 or 5 hours a day on it, just scrolling through, you know, great feeds and, you know, people that I knew and yeah, and it's just, it ate so much time up in my day that I just haven't been back. And then with all the bullshit they were doing with, you know, shutting channels down and everything else. But, you know, I still have some videos up. You can see my old DWC that I used to have going at that place that I show. And then my transition over to Cocoa. And then hopefully you'll see me transition over to some living soil here eventually. And, but no, you can find me either on your show. And I'm always honored when you ask me on, you know, allow me on there. Potent. I really appreciate it, man.
Okay, I love having you on.
Thanks, man. I really appreciate it. No problem. I hope everybody has a good night. I'm gonna head out.
Cool. Cheers. Thanks for coming.
Peace.
Cheers.
No worries, man.
We had a question from Chad. What did I say after Bavaria bassiana? It was Isaria, Isaria, Fumiceraceae. I don't remember how to spell Fumiceraceae off the top of my head. The new spelling is Cordyceps Fumiceraceae. So you can look that up. The product that is the two products that come to mind would be a product called Ancora, Anchora, I think it is, or something close to that. And the other one is PFR 97. So PFR 97, and you can find the species name from there. So either way, you'll get the right name. The Metarhizium is coming back. Metarhizium used to be a really good product called Met 52. It was cool because it also worked on ants, but ants are something that's kind of harder to treat. Same thing with termites. So that was neat, but they pulled it from the market temporarily. But now there's a new company making it, so that'll be coming back on the market the next month or two. So look for that. I don't know what the new commercial product name is, but I do know that it's coming. So, alrighty, everybody. Well, thanks for watching. Always love having Kevin McKernan on the show. Again, you can check him out at medicinalgenomics.com. You can check him out on Instagram at Kevin_McKernan, McKernan_73 on Instagram. His podcast is called Coffee Talk. You can find it at CannMed Events, CannMedEvents.com/coffee-talk. You can find out his the genomic sequencing for the psilocybin cubensis is going to be at Cilacida, so PSILOCYDIA.net to check that out if you want to see the sequencing on that. Yeah, there's all kinds of goodies. You can find myself and Marty over at APMJClass.com. Again, we have about 24 hours left on the class sale. Tomorrow is the last day of the sale for Earth Day. Again, AP 420 for $75 off the class. We have a full long length class on that. We'll also be launching some new classes later on this year on some specific topics, but definitely check out this class on the full length aquaponic cannabis. We kind of cover everything top to bottom. So if you're interested in that, check that out at apmjclass.com at ap420.com. Alrighty, guys. Really appreciate you taking the time to watch the show. Thank you, everybody, for all of our support over yesterday was 420. We're hanging out over on Fumidor's show. In fact, let me throw Fumi's site up on the thing real quick, just to make sure that everyone knows how to find him as well. This is his website, fumidoro.com. He has all kinds of wonderful photography and genetic preservation kits and more on there. You can find out over at fumidoroseeds.co is the name of the company, and fumidoro.com if you're interested in that. There's some wonderful things. Certainly the best photography I've seen of cannabis on the web. All right. Thank you everybody for watching. You can find our podcast on SoundCloud, YouTube, iTunes, Spotify, all the things. We'll be back again next week on Monday. I don't we will have a guest on Monday. We're not ready to announce it yet. Thursday we have Angela from Aquilitas from up in Canada. Really excited to have her on. She's got a bunch of new data to talk to us about on that. Bunch of cool new projects that she's been working on, so that's going to be super exciting. I think she's about ready to unveil some other cool new projects publicly that'll be cool new resources for you all to use out there. So that's going to be fun. And then we also have Tommy Chong. Tommy Chong is going to be on the show on May 12th. We're going to do a special time, 3 p.m. Pacific, because of his availability. So be sure to tune in May 12th. We're going to hang out with Tommy Chong. I'm super excited. I've been listening to Tommy Chong as a kid. My dad used to put on that, Tommy Cheech and Chong Christmas, and my mom would get all mad at him for putting it on at Christmas time. And we listen, and they talk about, the magic fairy dust, a little bit for the reindeer, a little bit for Santa, a little bit more for Santa. Anyways, all the goofy shit that they have on that record, the vinyl, that was like Christmas to me. It was like dad putting that on. So really, really excited to have Tommy Chong on the show. It certainly is like a bit of a milestone for us on the show as well. So super stoked on that. And we're going to have a lot of cool guests coming up on the show. I got some cool people lined up that we have. They're going to come on the show, but we're working on dates. So a lot of cool people, we're going to work on trying to get in some more scientists on the show, some more clinical people on, and really kind of step up the game on some of the guests on the show. We've really kind of been on a bit of a roll this year as far as guests and stuff. I think you guys would agree, and I think you guys can look forward to some really cool ones in the queue as well that we haven't announced yet. So thanks everybody for watching. Thanks everybody for the support over the last 6 years. It really truly does mean a lot, and I appreciate you all. Have a great night.